Well, I send you greetings from the East Bay Regional Parks today. I'm wildlife biologist, certified wildlife biologist. My credentials are on the board. But most importantly today, I want you to leave with a little PT, a little desire in your heart to protect turtles, to be educated, to be enriched, to be inspired, to help these animals. Some of the key points I'm going to share with you in the next 15 minutes. 15 years of multi-year data on male and female showed that male and female western pond turtles use a pond differently. So when you're thinking about restoration, you're thinking about reintroduction, you want to think about how the different sex ratios use a pond. I'll talk about overwintering, how turtles overwinter. I'll talk about how females who have eggs, gravid females, the length that they leave the pond, the type of habitat that they select to lay their eggs in. I'll talk about northwestern and pond turtles and females and their growth rates, population dynamics. And I want to stress at the end drought. And drought conditions are severe on turtles. They can starve. They're subject to dehydration, predation, and in 2021, this pond that I'm sharing with you today at Clayton Ranch, it was built over 90 years ago, was the first time that it dried up. And we lost over 52% of those turtles. And that's a significant number when you're thinking about a long-lived animal. It takes a long time to reach sexual maturity. So climate-induced stressors are going to be big on reptiles and especially on turtles. So with that, I'm going to get started. So hang on. Tell the person next to you it's going to be a rapid talk. Okay. So I want to thank the 7 different institutions that have helped fund this research over the last 1.5 decades. I also want to thank our wildlife volunteer team. Over the last 15 years, more than 60 people have donated upwards of 15,000 hours in our turtle telemetry team, putting their neck out to help the western pond turtle at that time when we started, now considered the northwestern pond turtle. So why do we study turtles? Well, they're cool, right? They're cool. They're reptiles. But throughout the world, they're facing many stressors, and you can see on the pie chart there's a lot of things that are affecting their survival. Reptiles, turtles, lizard snakes worldwide are declining. We want to support conservation efforts, as we heard about the Diablo Range. The western pond turtle is predicted by modeling around 2050 to 2075 to be extinct. And that's heart-wrenching. As a biologist, as someone who's interested in the environment, as environmental practitioners, to think that these incredible animals that have been on the face of the planet are going to go extinct within our lifetime. So we want to stop that. All right, so why the western pond turtle? It's our only native turtle. You have the southwestern pond turtle, you have the northwestern pond turtle. They're both found in the East Bay Regional Parks. They're candidate for listing under their federal. It's moving along at a slow pace. Pun intended there, at a turtle pace. California species of special concern. It's been a species of special concern for over 30 years. It's been listed as threatened in Oregon, endangered in Washington, and it has special status with the Forest Service and the BLM. So a lot of legal aspects of why we're interested in the turtle. So why the decline? I've got 2 pictures here, and these will be on the exam for lunch. Okay? Habitat degradation is a big part of that. Humans are a big part of that. The person next to you, we all have a role to play on the decline of turtles. Certainly this picture right here, this widely introduced turtle, the most noxious of introductions, ranks in the top 100 of invasive species, has been spread by humans. And this, of course, is the red-eared slider. Also, non-native predators, bullfrogs and largemouth bass. Non-native fish eat lots and lots and lots of turtle hatchlings. Overabundant raccoons, skunks, and rats do a number on nest sites, do a lot of nest depredation of turtles. Epidemic disease, respiratory issues for turtles. So there's a lot of reasons why the turtles are declining. Just can't put your finger on one thing. There's a whole bunch of parts in this puzzle. So what do we know about the ecology of turtles in Central California? Not a lot. There's a research gap. I'm going to go through some of this really quickly. First of all, up by Nick Geiss's group, in Sonoma State, and you'll hear Maddie later on today, and I'll leave her thunder for then. But nest site fidelity in a semi-permanent pond, this is north of Alameda and Contra Costa County. Response to drought in the Sierra foothills, so this is far east. And as I already said in my introductory notes, drought is not good for turtles. And in that situation, the turtles, high predation, they moved into the uplands, a lot of turtle loss. Terrestrial movements in 2 ponds on the Creasville Plain. So in this research south of our location, turtles would move into the upland shrub to survive the heat. But in all 3 of these sites, there's no active livestock grazing, and that's my point. The talk I'm going to talk to you today about is about turtles living with livestock. And lastly, growth and population by Germano, big turtle researcher. But this is on the coast, and the growth patterns of turtles on the coast are different from the growth patterns of turtles here in Alameda and Contra Costa County. So as I said, there's a variety of life histories in turtles. And we get a lot of emails from the regulators and the recovery team asking, what about this? You can't just say one size fits all with a turtle. If you're living in a pond, you do one thing. If you're in a seasonal pond, you do another thing. If you're in a river, you migrate out. So there's a lot of plasticity, I'll say, that creates challenges for trying to come up with a conservation strategy. And as I've already said, one size is not going to fit all. One glove's not going to fit. So a lot of different approaches. You have to have local research to help guide that decision-making process. So research gap, and what I'm going to share with you today is northwestern pond turtle in a pond environment that's subject to livestock grazing. So this is located in the beautiful Clayton Ranch, just southeastern side of Mount Diablo. There's Marsh Creek Road. There's the pond, Turtle Town, a coup de grace of turtles in the Park District in eastern Contra Costa County. All these ponds have livestock grazing. All these ponds provide critical habitat. And these codes here, that's the northwestern pond turtle, that's the California red-legged frog, and CDS. So we have California tiger summit. So we have 3 listed species living in these ponds. And I want to work on this pond right here, and I'll make some introductory notes about that later. We have turtles that have moved into that pond, but that pond is blown out and then dried out. So if you have extra money, you can throw out a great restoration project. This is managed by East Bay Regional Parks, Alameda and Contra Costa County, the largest urban park district in the nation. And with that, a little bit more about Clayton Ranch. Annual grasses, I said to a couple of people, this is a beautiful location. Trees only comprise about 10% of the park. Both giving of the trees that are there are our oak trees, our blue oak, our valley oak, our imperial live oak. And if any birders in here? Okay, so the birders know over 174 different types of birds out here. This is a great spot to come look for birds. And cattle protection occurs on this site. This site is not open to the public. And I'll just answer the question now because I always hear it whenever I talk to groups. Have I ever observed turtles being trampled by livestock? The answer is no. In 15 years, I've not seen a turtle depredate a cow depredate a turtle. I hope I didn't say that reverse. That would be a very moving situation, wouldn't it? All right. Sorry, I'm trying to keep on track here. All right, so what are the objectives? All right, we want to document the movement patterns in a pond environment subject to livestock grazing between male and female turtles. We want to look at their habitat use patterns. How are they using the upland? How are they using the pond? We want to follow pregnant female mama turtles, describe where they're going to go lay their eggs so we can use management to help protect those sites. We're going to report on 15 years of market recapture data and growth turtles and the response to drought. And one of the things I was going to say, and I was mentioned to Maddie early on, is you get started in turtles when you're young, but they outlive a biologist's career because we're talking about animals that go 75 years. You mark them now, and your grandkids are going to be looking at them. And then I want adaptive management recommendations for conserving turtles in eastern Contra Costa County. So hang on. Turtle trapping, there's a lot of different ways to trap a turtle. We use floating sun decks and solarium traps. The beauty here is you have a bait station. Turtles love protein, okay? So you bait it with protein, and also you've got a nice spot when they have that sleep coma, like maybe after a Thanksgiving dinner or something, you get nappy. So the turtles like to climb up on these little ramps and sit there and sunbathe. So we got two things, food and a great spot for them to nap. So they fall in the trap. And then handling turtles, the male is on my left, okay? And I want to go through a little bit about secondary sexual characteristics in turtles. Turtles don't really show secondary sexual characteristics till they're about 5 years old, when they're over 120 millimeters in length, so that's about 5 inches long. So we have a male here in my left hand, and you can see that he has a concave plastron. His tail is thick, his clavicles way past the margin of his shell. Males tend to have a very pointed snout here. Let's see if I can get that, okay? Whereas the females, on the other hand, that was a pun intended, on the right hand, okay, is her tail is very thin, her plastron is flat, and her clavicle is not quite past to the margin, and females tend to have a more blunt snout and tend to have flecking. And this is a great slide here because it talks about aging. They have growth rings on them. You can see that this turtle still has her growth rings showing, and this old male, it's all polished off. So once they start to lose that turtle's well over 15 years old. So this is a big daddy. So handling turtles, standard measurements are used for turtles, looking at their weight, the length of their carapace, their plastron. One of the other things that we want to do is mark all the turtles. So we use the bury system. And you can see one of my former interns here doing the filing action. We can mark up to almost 7,000 turtles using this method. So when we catch it, we can still look at that filing mark to tell which turtle it was, to get individual recognition. Then all turtles were fitted with ATS transmitters that weigh 12 grams. It was glued to their scute. That would take up 3 to 5% of their body weight. And the antenna was so it wouldn't hang up during reproduction. You don't want somebody grabbing onto it and holding onto it when they're trying to reproduce. So now the telemetry. We had a beautiful grid set up of the pond, much like bird spot mapping. So we had stations across so we could spot where the birds, not where the birds were, turtles were. Sorry. I deal with a lot of different things, all things wild, so I got to remember which talk I'm talking about. This is not black rails today, not bats, it's turtles. So they were all followed using ATS model receivers. These are my daughters when they were teenagers. It was like, if you want to eat, you got to help Dad go study turtles. So we'd watch these turtles in the fall and winter to figure out where they're going to hibernate, where they're going to spend the winter sleeping. 3 to 5 times a week during the rest of the year, we'd follow their movement patterns. And as we got closer to the breeding season, the time when they're going to start to lay eggs, May through August, it was almost a daily event. Sometimes 2 teams, a team in the morning, a team in the late afternoon, to follow these turtles. Now, this is Richard, one of our wildlife volunteers. I mentioned volunteers are a key part of our work. And this was one of the longest turtles, a gravid female we followed. And she is right there, okay? She's right there. So during the evening, we followed 40 females from end of the May through July every year, figuring out where they're going to go lay their eggs. We collected GPS coordinates of all the nest site attempts. 1 minute left? Okay, I'm going to speed this up. Measured RDM levels of each site. I'm going to have to go over a couple minutes, okay? We'll skip the song. See me at lunch, I'll teach you the reptile turtle song. Okay. Or come in the field, I'll teach it there, okay? So what I see here is that this number right here, this is a wonderful p-value. Males and females are not using the pond the same way. Okay, some turtles we know from the literature stay in the ponds all year, some move on to land. Our turtles overwintered in the same location right here at the mouth of the inlet of the pond. So here we got the females, and all the females are found in this grid for their primary proportion based on the stats. Typically, This bank here was at a 1-to-4 ratio, so it's a 14 degrees. And that's important if you're going to start building a pond, you're going to start dealing pond restoration. This has to deal with the energetic demands of a female turtle. They can't climb as well as a male. Okay? Now let's go to the boys. And the males were not found in the same locations too. And most of their locations were where there were great rocks and basking spots right here, or where the females were. And that makes sense, right? Because turtle movement is going to be related to where you can find food, where you can reproduce, where you can bask and hide. And this location here was at a 2-to-1, so the males needed a steeper slope, selected a steeper slope. This is the turtle locations. All females use the south-facing side at those degrees. They moved an average of 24 meters away from the pond. And oviposition egg laying typically occurred at 4:30 in the afternoon, and the same females returned to the pond that day. So what are some of the key aspects? Seasonal grazing. The RDM levels is right 13. That's biomass, a grazing number. Annual grass height was about 15 inches. And this is interesting because it corresponds to a lot of our lizard work that we published early on, similar things. Trees were not important. Okay, so what does it suggest? That males and females use the pond differently. However, they hibernate in the same location. There's a difference between the way males and females. The females were using a more gentle slope, males a more steeper slope. Females would leave the pond at these 3 locations because it's less of a slope, and they went to lay their eggs in locations 24 meters from the pond, and it was like a mass exodus. The last week of May, the first 2 weeks of June, that's when all the females, all 40 females, would leave the pond to go lay their eggs. So that first week of June was like the key time for turtle movement to go lay eggs. And good sun exposure. So this is growth data. Blue dots are males, the green are females, and you could see that initially growth is very fast growth rate the first 10 years. And this was really a nice significant value that males grow faster than females, and that supports the literature. When we look at the carapace length between males and females, while it's important, you can see in the birding reptile guides that the males are definitely larger, and we can see that it wasn't statistically significant. So that's one of those things where stats and biological things don't line up. Now this is where I want to spend the last 30 seconds if I can. Okay. This is all about drought, all right? And you could see that the drought that happened in 2021, the first time that pond had ever dried up in over 85 years from building, you can see we had over almost 50 turtles marked. Only half the population survived. I mean, half of the population perished. That's a significant thing when you're thinking about a long-lived animal, okay? And then you look at this, you'll notice that there's a definite sex ratio difference between males and females. Females outnumber males 3-to-1 in that pond. And we look at this, there was no statistically significant difference of female survival, but it's suggestive at the 85% level, not the 90% level, at the 85% level, that there's definitely a difference between male survival in a drought situation, a turtle. So the conservation management is baseline, helping us to provide a framework to better understand turtles, dynamics, how they use the uplands, the aquatic environment, growth rates, the response to drought. And this information is going to be used to help develop a robust turtle plan. So some of the next step is to look at how turtles respond to pond restoration, following some of our work on red-legged frogs. We want to, and certainly the career that I have left, look at nest depredation and look at ways to improve wild turtle hatching, and look at some of the thermal biology. How are those males and females thermally selecting the pond? A couple of publications you can look for. And I just wanted to say that I hope you were inspired. I hope you got some great information. I hope you'll stick your neck out for turtles now and think about the habitat loss invasive species and the effect of drought is going to have catastrophic implications for the future of the northwestern pond turtle in the Diablo Range. Thank you.
Dave, you alluded to the need to restore the pond. You've got two adjacent to Marsh Creek Road. The upper one appears to have a blown out spillway. What is it going to take in terms of dollars and push that project forward?
To push that project, that's a great question. I'm glad you asked that one. That's the pond I want to finish before I leave. Right, Doug? Right. So that's going to require about $35,000 to $40,000 for materials to make it happen. We have a crew of engineers. Our roads and trails crews have already done that kind of work with our ponds for red-legged frogs and how to do it. We need permitting, and we need an engineer design. That's what it takes to get it done. And we're trying to get that done in the next 1 year.
Would you have Doug bring that up to the Mary Bowerman committee at our next meeting?
He's on it. Okay.
Are there any restoration strategies that would allow livestock and the western pond turtle to cohabitate?
Yes. I'm sorry I didn't clarify that. The western pond turtle and the livestock in this pond are cohabitating wonderfully. The livestock get access to the water. One of the things that we've done with the grazer is once we determined when the females were leaving, I asked him to use the water sources away from the pond. We never saw any evidence in 15 years of turtles being trampled by cows in the ponds. I haven't found any hatchlings that have been trampled. A hatchling takes about 8 months for it to hatch, stays around the nest site for 7 to 9 days, and then migrates down to the pond. So there's definitely strategies where livestock, and we're modeling it now. That's all this information kind of feeds into that. Wonderful question. Thank you. Thank you, Sean. Sorry I went over.
Hi, thank you for having me. Here we go. My talk is on the Flora of the East Bay, a new book by Lech Nelmovich and Barbara Erder. My purpose here is the purpose of the project actually is to update a 2013 checklist of East Bay vascular plants, which itself was the second edition of a 2002 checklist. The authors renamed the book Flora because of its scope and purpose, which is to serve as a thorough and essential reference guide to the plant life of the East Bay. To give some background, earlier this year, Holly Forbes of Save Mount Diablo knew that Lech had begun work on this project and that the California Native Plant Society East Bay chapter was fundraising for it. So she invited us to write a grant for the for the Bowerman Science and Research Grant Program. And so thank you. As an organization dedicated to native plants in Alameda and Contra Costa counties, CNPS East Bay chapter has always been deeply involved with providing information about our local native plants. My role has been project manager, copy reader, and cheerleader because as a botanist, I use local floras all the time, and I know how valuable they are both for professionals and for amateurs. Why am I here? Well, the authors live in Idaho and Montana, so they send their greetings. Yeah. So I'm going to start with some highlights, tell you how the flora is put together, why the flora is a useful tool, and how you can get involved. So first, this is Barbara Erder, the creator of the East Bay checklist, and someone you already know as, oops, how did I do that? There we go. Co-author of the updated Flora of Mount Diablo, which you all know well. This is Lech Nelmovich, who was co-author on the last checklist, and now is the senior author on the Flora. So here's some highlights. The Flora has updated information on all East Bay wild growing vascular plants, with some plants added and others removed, dozens more reclassified, and thousands of records evaluated to add or correct to the distributional information. It has a new list of bryophytes. Those are non-vascular plants, which include groups of mosses, that's the most common, liverworts, and hornworts, all tiny little plants. It includes more information about common names, status, terminology definitions, and most helpful for people like me, more information about how to distinguish similar plants from each other. Fun facts, the Flora has listings for 2,052 vascular plants, a net increase of 42 since the last checklist was prepared. Of these, 1,329 of them are native taxa, and the other ones are 557 non-native species and 166 that are called waifs. Waifs are plants that are not indigenous here, but are growing independently, that is, in the wild without human management or care, but are not known to be reproducing either by seed or vegetatively in the wild. We've got 211 bryophytes, of which 210 are native. And yeah, we have one non-native on campus here, so thank you. So the East Bay supports 20% of all the California native plant taxa in just 1% of the land area. So I'm just going to give you a moment to let that sink in, because even though when you look across the landscape, you see a lot of non-natives, we have a remarkable native plant diversity. Some species are at the edge of their range, some are uncommon or rare, some are here and nowhere else. Mount Diablo, of course, has some of our best examples, but we have many others, often in very specialized habitats. So even in an area that has a tremendous human population and is well studied botanically, changes in knowledge continue to take place at a actually fairly impressive rate. So now I'd like to talk about how a flora is put together. Originally, the authors combed through herbarium specimens, the physical, actual herbarium, floristic studies like the flora of Mount Diablo and others, statewide floras, technical reports, and CNPS records of rare plants. Herbarium records are now data-based, so electronic data can be searched for new records. Also, citizen science reports are available through sources like iNaturalist and CalFlora. And of course, there's a shout out to the many, many contributors to knowledge, especially Diane Lake of the Native Plant Society's East Bay Unusual and Significant Plants team. They go out and systematically monitor known populations and search for new ones for some of the least common plants. But many other people contribute information as well. So once these mountains of records have been amassed, begins the serious work to evaluate them. Botanists are only happy when they have a physical specimen to document a plant record, because many diagnostic plant features can't easily be photographed or documented in other ways. A physical collection like this can be examined and re-examined by experts, and specimens also provide samples for genetic work. As a result, herbarium collections are considered the gold standard, but reliable botanist accounts were readily accepted, and less well-vetted records were examined carefully. The more unusual the record, that is, a report of a species well outside its range or in an unusual habitat, the more critically the authors evaluate it. Sometimes these reports were accepted, sometimes they were rejected, but the authors kept records of all these so that with new information, they can be reevaluated. Keeping up with new records is one part of the challenge, but the other is keeping up with the science. Huge changes in plant taxonomy have taken place at all levels since genetic analysis has become an essential tool. Fortunately, the UC and Jepson herbaria continually update California nomenclature online with the Jepson eFlora. The next level of detail is keeping track of plant distribution. New records are eagerly added to the flora once they've been vetted, and erroneous records are just as eagerly weeded out. But to best describe the distribution of plants in the East Bay, the authors created these 8 bioregions. These are areas that are ecologically similar enough that a plant found in one part of the bioregion might reasonably be found in another part of it. I don't know if I can do this pointer well enough to see, but so far on the left is the Molati region, the smallest one with some coastal prairies. The flatland region runs along the bay. And then we've got B for the Bay Hills, B for the valley section up along the Carquinez Strait and into the delta, D for the Diablo region. L is the Livermore region in this Tri-Valley area there. Hamilton Range is down, it's actually the dividing point is Niles Canyon right there. But this is the more eastern and higher mountains to the south and east. And then the Altamont is the mostly treeless area in between Livermore and the Central Valley. So here's some ways that the flora helps East Bay knowledge. One is keeping track of new introductions. LEC feels a special responsibility to keep up with introductions of new species to the East Bay because some become invasives. By putting out timely information in the flora, even if they're not in the Jepson manual, users can become aware and potentially manage invasives before they get out of hand. And even though I was mostly sort of one step away from the authorship, I did make a little tiny contribution here. This is Carex divulsa. This plant has been mistakenly sold in the nursery trade as a native sedge. So once I discovered it was in my native plant garden, I spent the next several years getting rid of it. But yeah, it was a lot of work. Just being in a garden, though, was not enough to qualify it for being in the flora, because the flora has to be, it only deals with plants that are growing in the wild. So we searched CalFlora and found a record by a very reputable observer in Wildcat Regional Park. So that was our latest addition to the flora. So keep an eye out for it, though. Another function is monitoring plants that are on the edge. Our 2-county area supports 21 federally and state-listed plants, 124 plants that are ranked by the California Native Plant Society statewide, and about 900 more that are considered unusual and significant in the East Bay, meaning they're at the edge of their range, thought to be declining in the East Bay or elsewhere, and even relatively abundant elsewhere, but uncommon here in the East Bay. CNPS's Unusual Plants team monitors hundreds of records every year, but the flora helps the user see if they've encountered an unusual sighting of some sort. This is our iconic pallid manzanita, a species that is completely restricted to our 2-county area and only on marine sandstone habitats. This is a happy story. Plants that are thought to be extinct are a special case, but tracking them sometimes yields exciting results. This tiny caper-fruited Tropidocarpum was thought to be extinct since the 1950s, but it was rediscovered this spring by an astute botanist. So we updated that in the flora. And then the next thing, you received a handout on your seat, or you might have picked it up coming in, but there's so much detail that I wanted to share with you, and it was just going to be impossible to see it on the screen. So grab your sheet of paper. The front page is the, oh, thank you. The front page is the colored page, and it shows you the information contained in the vascular plant section of the book. It's color-coded, so it's a little easier to see things, but the I on the first column in white is introduced plants. Yellow is cross-references, so something that you might have known as convolvulus in the past. The native species have been reassigned to a genus called Calistegia, so that shows you where to look for that, for the name that you used to know, and now it's in a different place. W is that peach color, the waif, and that's a species that is actually not native here, but apparently not persisting in the wild or reproducing. And then I picked a page with a lot of native plants. The green shading is native plants, and with an asterisk is an unusual plant. So the next column shows you the scientific name, family affiliation in brackets, common name if there is one. And then the next column is the rarity status. And again, I picked a page with a lot of examples here, but the ones with a U means that they're locally unusual or significant. A C means that it has a state rank, and if there was a federal or state ranking, that would have another code. The next column is the codes for distribution. So B would be Bay Hills, D, Diablo, H, Hamilton, and the number next to it is the quality or reliability of the data. And one thing that the authors are interested in is if there's something that's not a 1, which is the gold standard, a herbarium specimen, then they're interested in documentation of some of those less reliable records to document them for sure. The next column, this little small column, is the life form. W is for woody, H is for herbaceous. Other categories are grass, aquatic, and ferns. And then the big column on the right-hand side is just information that you might want in the field, telling you how to distinguish related species. And believe me, I love that part. Okay, so then I just wanted to say one other thing. Let's see. LEC had long wanted to include a list of bryophytes because these are truly a new frontier for many botanists. And LEC wanted to raise awareness to make it easier for people to get to know these fascinating but poorly known plants. And I don't know if she's in the audience, but I just want to give a big shout out to Katie Colbert, who assisted in this project by methodically reviewing all the available publications on California bryophytes, compiling name, synonymy, distribution, and habitat for this book. And so to turn over the handout, you can see the information on bryophytes is a lot more sparse. So we need to populate that, thank you. This page shows you, this is a moss page, so that first column, the M's mean that it's a moss. Scientific name, brackets for the family, and then common name if there is one. Rarity status, and this is pretty typical. We don't have enough information to really assess rarity status for most mosses, liverworts and hornworts. So there's just one here. The next one is distribution, and most of the distribution is not well known. Mount Diablo has been well surveyed for bryophytes, so the MTD means that this plant has been seen on Mount Diablo. Sometimes we have some bioregions, B for Bay Hills, H for Hamilton. Sometimes we only know the county, and so there's some codes for Contra Costa and Alameda counties there. Bryophytes are funny. They don't really respond to plant communities so much as substrate. So the code in the next column tells you that S is a moss, in this case, that grows on soil. R is for rock, T is for trees, and SR is a sort of a specialized soil over rock, which many mosses inhabit. And then you can see that it's kind of brief, but the next column just shows you what information is known about sort of microhabitat or distribution. Let's see, oh, we already did that. So what you can do, well, grab your field equipment, go outside, make some observations, and share them with us. We have a special website, a special email address that you can send observations to, and I have already started marking up my proof of the new book. So send us some information and share it with us. Thank you. And finally, I just want to say thank you to the Save Mount Diablo Mary Bowerman Fund for allowing this and supporting this work. Thank you.
When are the books available?
Oh, thank you. So they're at the printer, and I'm going to pick up the first sort of couple of boxes of them, and they'll be available at Native Here Nursery, which is the Native Plant Society's native plant nursery at Pentilden Park tomorrow during open hours. And then if you look at the back page, you'll also see that there's a website that you can order books and have them mailed to you, or you can order them and pick them up at the nursery. But tomorrow and Monday for the rest, yeah. Anything else? Yes, in the back.
Sort of a more general question about distribution of plants in the region. I'm wondering if anyone in your group or overall the botanical community around here has noticed species becoming more common that were more typical of the drier, hotter places in the south, moving north. And I've also been told that several different organizations are actively restoring habitat using species more typical of the south around here to sort of like get ahead of climate change and things like that. Any trends like that?
Wow, I don't think I am prepared to answer that question. It's certainly been a topic under much discussion and thought. I think that we want to be careful to think carefully about what we're doing if we're moving things into habitats where they didn't exist before. I mean, probably a more conservative way of approaching might be to look at local genotypes and possibly think about those moving rather than species. But yeah, it's all, it's what we have to think it through because climate change is happening and we have to figure out how to best protect our resources. I was going to say one thing though, this is kind of cute. When the announcement came out for this colloquium, I received an email from a colleague who works primarily in the Central Valley and out of the Modesto area. And he said, wow, great, you got an East Bay flora. And he was aware of it. But he said, like, how about a flora of the Diablo Range? I've got these plant lists from, all these San Joaquin, Stanislaus, Merced counties. Let's collaborate. So just saying, there's some big interest there. Yeah.
Of the 46 additions to this checklist, do you have a breakdown of what were like newly introduced species versus range extensions or like newly described species or taxonomical changes?
Well, we had, so roughly 1,300 species are native taxa, and roughly 550 are non-native, but are established in the East Bay. They're reproducing and are well established, and then 166 are waifs. I think I went by it pretty fast, but I don't know if this is answering your question, but we do have some, there were net changes of a couple of dozen species of natives. And that's, I mean, let me just say, I mean, I didn't do all the work that it took to get this. I'm not the author, but that's a net change. So some species got booted out, some got brought in, and the net change was a couple of dozen native species and several dozen non-natives. So they're always tracking them. And information changes because there always are new records within our 2-county area.
Yeah, I was just more curious about the actual update compared to the last checklist. That's
Yeah, those are numbers since the last checklist. Yeah, and I guess another thing I didn't mention, and this just has to do with more eyes, more people knowing that there's something they're seeing that is noteworthy or could be noteworthy, is that the first checklist was something like 1,600 taxa, and the second checklist was something like 1,800 taxa, and here we are with the flora, which is essentially kind of a third edition, that's got 2,000, and that's partly because there are more introductions, really, for the most part. There are some discoveries of things that people didn't have records of that were nearby, but now we've got them in the East Bay, and of course, new introductions. So the state of knowledge is always going to add more complexity to what we know.
Thank you so much. I'm really happy to be able to give you a quick update on our long-term ground squirrel research at Briones Regional Park. Today, I'm specifically going to be focusing on so-called ecological ripple effects that affect squirrel survival and behavior after a vole population boom. Now, our long-term ground squirrel project was established by Jen Smith in 2013 during her time at Mills College. She has since moved to a position at the University of Wisconsin-Eau Claire. So since 2023, I've been co-leading the project out of UC Davis, and I've been running the field seasons together with the many undergraduate students that come through and help us monitor the populations and just generally support the research. Now, we've been live trapping the squirrels with peanut butter and sunflower seeds since 2013. Once trapped, they each get a series of marks so we can recognize them over a long time. They get a little metal ear tag, a so-called pit tag, which is basically just a microchip under the skin, similar to what we sometimes give to dogs and cats. And they each get a little paint mark on their back, so we're able to visually identify them with binoculars in the field. From each squirrel, we also collect a small piece of their ear tissue from which we extract DNA, so we can estimate relatedness among the squirrels. We collect ectoparasite loads, so we literally count how many fleas and ticks they each have. And we regularly collect fecal samples from which we can extract so-called glucocorticoids, which are also called the stress hormone. So this gives us an idea of how physiologically stressed each individual is. We also regularly collect or conduct behavioral observations where we record all sorts of social interactions that allows us to build these social networks so we better understand who these squirrels preferably interact with, who they hang out with. We create spatial maps of where they preferably spend their time. And we also record all occurrences of humans, of dogs, of natural predators across the site that include coyotes, rattlesnakes, birds of prey. So we get a really detailed understanding of their environment. And the breadth of the data that we collect also really reflects in the sort of research that comes out of this project over the years. For example, work on anti-predator behavior. How do they read predator cues? Their social and spatial networks, or how stress physiology affects venom resistance against rattlesnakes. So just to show you a few examples. But what I want to focus on today is the past couple of years. You'd think that after studying the same population at the same place for over a decade, there are not so many surprises left, but 2024 really had a novel one in store for us because when we came to the field site in summer 2024, we found it crawling with these little critters, which turned out to be California voles. And this species typically undergoes these population cycles where they peak every few years in response to favorable weather conditions, food conditions. So that part was normal, but just the sheer numbers in 2024 were incredible. So this is data that we downloaded from the citizen science app iNaturalist, and you can see how enormous this population peak was in 2024. And if you live in the area, you may have seen them running around during that summer too. Geographically, there were several spots that had these population booms, but it was definitely most pronounced here around the Bay Area. But this is not where the surprise ended, because one day, two students approached me with this video of one of our squirrels seemingly attacking one of those voles. And at first, I wasn't really sure what to make of it. I had not seen a behavior like this before. But it soon became apparent that their squirrels started hunting those voles. And over the next few weeks, we recorded over 70 occasions of squirrels attacking, killing, and consuming these voles. And some of them got really good with time, and the behavior reminded more of a cat or some predator, like hunting and chasing after them, and often like killing them with a bite to the neck. So it was quite spectacular to see. Once caught, they usually consumed the entire vole. So that is a really high nutritional diet compared to their normal seed eating or eating fresh greens. Most occurrences were in early July, which also coincided with the peak in abundance of these voles at our site. We saw the behavior pretty much across the entire study area and in members across all sex and age classes, which suggests that the hunting is actually an innate behavior that emerges in response to increased prey availability. And that really just highlights how opportunistic these squirrels are in their diet and how behaviorally flexible. And that's definitely partially responsible for their enormous ecological success in them invading all sorts of different habitats because they're so adaptable. The story was also extremely popular in the news. The world loved the story of the killer squirrels in the bay. Are we next? So you might have seen it somewhere on the news. Now, this is really new data. We only just got this in about a week ago, because turns out the voles were not the only victims. From the fecal samples that we collected, we extracted DNA to get an idea of other prey items they might have been consuming, and about a little over 20% contained vole DNA, but we also found pocket gophers, several mice species, and even salamanders. And this again highlights that they will happily incorporate animal protein into their diet when opportunity presents itself. Now, naturally, we were very curious about the consequences of this increased nutrition during the summer, because this is, as you can imagine, this is a really high protein diet. Typically during summer, these squirrels put on a lot of weight in preparation for winter when food is not as abundant. So we expected that overwinter survival might be increased, as well as the reproductive rate next springs, because squirrels in better body condition usually have more or larger litter sizes. So coming back in 2025, we're really excited to be seeing loads and loads of squirrels, but that's not what we found. It was an absolute ghost town. And the numbers suggested that we had a devastating population crash of about 80% in our squirrels. So what happened? It became soon apparent why, because we also recorded record numbers of coyotes, rattlesnakes, and birds of prey around the study area. So what we were likely dealing with is a so-called ecological ripple effect, where the increased vole numbers have led to an increase in predators that then, once the voles died down, had to shift to a different prey species, the squirrels. So Normally, an adult squirrel in our population has about 60 to 70% of being recaptured the next year. And from 2024 to 2025, this dropped to about 20%. And this was even more extreme for the juveniles. Of more than 50 juveniles that we named in 2024, a single one survived till 2025. And that is not atypical because juveniles are typically more vulnerable to predators since they need to learn how to evade the predators, how to recognize them, and also because the juveniles are less resistant to rattlesnake venom. And this trend continued in 2025 during the summer. We captured a lot of females that were lactating, so they had pups somewhere in the burrows, but many of them already got eaten presumably by rattlesnakes before they even managed to emerge from the burrows. And even when they did emerge, they often got taken by some predator even before we had the chance to trap and mark them. So the entire 2025 summer, we marked one juvenile that we're obviously all rooting for, so hopefully he made it. Now, when faced with difficult changing environments, many animals, including us humans, adapt their behavior to kind of adapt to the novel conditions. And that's something we also found in our squirrels. We often conduct so-called flight initiation distance trials as a measure of how reactive they are to humans. So we literally walk up to a squirrel, and then we record the distance at which the individual runs away. And turns out these squirrels are very consistent in their responses. So a bold squirrel will always let us walk up a lot closer than a shy squirrel who will always run away at larger distances. So this gives us a measure of boldness. And what we found is that they substantially increased these flight initiation distances in 2025, suggesting that they were generally a lot more cautious about their environments presumably because of this high predation environment and also because of low squirrel density, because if there's fewer squirrels around, the pressure on you as an individual is a lot higher, so the likelihood of you getting attacked is a lot higher. So they generally increased their levels of caution. Now, such natural disasters are obviously really devastating for a population, but bottlenecks, which we call them when a population gets reduced to just a few individuals, are also really exciting from a science perspective. And they're extremely difficult to study in the wild because, of course, we never know when or where such events might happen. And this is where long-term projects like the squirrel projects are extremely valuable because we have this detailed data available on individuals, on the environment, both before and after the event. And that allows us to exactly study the process of recovery. So the squirrels, of course, they experience this really devastating population crash, but also they are resilient. So I'm assuming they will recover in time as the predator numbers will inevitably go down when there's less prey available. So this gives us really the chance to kind of document this recovery process, to look up, for example, if there's shifts, like who survives and who reproduces. Are there, for example, shifts towards more shyer squirrels because they are able to better evade the predators? Are they the ones that are more likely to reproduce? How does the dramatic reduction in the gene pool affect the diversity of the population if only a few individuals then kind of found the new population? Also, how do they spatially recolonize the area when there's so many empty burrows available? How does it affect the juvenile individuals' decisions of where they will establish their home ranges when they move away from their natal area? Also, how do they change their behaviors in relation to these fluctuations of the predator numbers? And also, how does the low squirrel density affect parasite dynamics? Because obviously you can't transmit your parasites if you don't encounter anybody. And that has important implications for the diffusion of illnesses. So that has important health implications. So we really cannot wait to get back out there next summer to see what's been happening, and of course to see if our one juvenile that we named is still alive. So I really want to thank my co-leader of the project, Jen, who's been managing a lot behind the scenes. And then, of course, many students have been coming out over the years to support the project. A special shout out to Team Squirrel 2025. Because of these really low squirrel numbers, they had to work extra hard for every single data point. So they had to really show a lot of patience when we just didn't trap anybody for hours and hours on end. So they really did work hard in a whole different sense. And of course, I really want to thank the Mary Bowerman Science and Research Program for their financial support towards the housing of the students that joined from Wisconsin, which is a direct contribution to their education. So being able to participate in such a field project really opens so many doors for these students. We're able to write them really great recommendation letters, and they have been really successful on their future career paths. So thank you very much.
So you mentioned in 2025 the population plummeted. They had an increased flight distance. Did you see glucocorticoids in the feces, higher levels of it?
Very good question. We did check, and weirdly enough, we did not see any changes. And we're a little bit puzzled because you'd think that there would be a general increase, but seemingly they didn't respond by increasing the glucocorticoids. No. Mm-hmm.
Great talk. Do you know if squirrels are predating different animals in different habitat types?
Very good question. From what we know, we work with two adjacent populations. The one that is like right next to a parking lot, there's a lot of like picnic tables, one that's a little more pristine further up the hill. And we were curious to see if we found the behavior in both areas. And it seems that they were predating on the voles pretty equally. And my guess is that but we don't have the exact same behavioral observations available in that second population. But judging from the DNA that we got out of the fecal samples, they will really just eat what's around. And one of the samples, it was a little bit low quality, so I didn't include it here. There was even DNA of a wild boar on it. So clearly there was a carcass somewhere, and they were like, well, thanks for the meal. So very highly opportunistic.
Hi, do you have any historical diet data to compare the introduction of voles?
We do not. No, this is not something we've looked at before. This was a thing we were like, oh, this would be fun to look at, but we don't, unfortunately.
Hi, do you have information on invertebrate prey as well?
Not at our site, but it is definitely known from the literature that they will quite regularly actually hunt insects. They will hunt small reptiles. They will predate nests, so they'll eat eggs. They will eat nestlings. So yeah, including invertebrate prey for sure. Mm-hmm.
It's super cool stuff. You think they're capturing all these prey items on the surface, or are they like diving into burrows after things? Because there's talk of California ground squirrel being a competitor with kangaroo rats in the San Joaquin, and that could be, an avenue for them to get K-rats.
Definitely. We think it's both. So what we often see is when the voles were really abundant, they were literally just kind of like walking up to a vole and catching it. The voles clearly did not perceive the squirrels as predators. But after a while, you could tell the remaining voles were a lot more cautious and would often try and go underground. The problem was that they often used some of the empty squirrel burrows. So the squirrel just followed them inside. And we've seen occasions of the squirrels running after the vole and then coming out with the captured vole. So they definitely do also predate underground.
There's quite a controversy with landowners, especially in agricultural lands, and overpopulation of ground squirrels in some areas. Do you think that any of the data that you're collecting can be used to help landowners successfully control ground squirrels without using rodenticides?
That's a very good question, and it's basically my end goal of my entire research. I mean, like, how can we mitigate this clear conflict there is? Because obviously, as an animal lover and excited about the squirrels, I'm very sad about them being culled in such large numbers. Some of the work I'm doing is looking at how they learn to access human resources. So that's directly related to them crop raiding, breaking into garbage bins, and so on. And I'm trying to figure out what in an individual predicts how successful they are. Is it behavior that they learn from one another? And can we do something that we, for example, instead of trying to kill them in large numbers, can we achieve something by removing target individuals that kind of spread this damaging behavior, for example, that others learn from? Has been successful in other species, but we're definitely at the very early stages of this in the ground squirrels. But it's definitely something, the research I'm doing, I definitely have that in mind of like a kind of an end goal. Yeah.
Awesome. Thank you so much, Sonia. If you have any more questions, make sure you ask her afterwards. Thank you.
Thank you.
Thank you all for being here. As mentioned, my name's Rory, and I am our Director of Conservation at Fresno Chaffee Zoo. And I'm here today to tell you about some work we've been doing for the last 5 years with these lovely guys, blunt-nosed leopard lizards. There's at least anecdotal observations of ground squirrels going after them also and chomping on them. So they may be yet another victim of the rabid killer ground squirrels. But today we're going to talk about some of their other threats and some of the things we're doing to try to bring them back throughout their range. To kick off, I just wanted to sort of point out this whole ecosystem that exists in California that not everyone's aware of. It's the San Joaquin Desert. It's a whole ecosystem that only occurs in Central California and has been about 95% depleted in the last 200 years. So that great Central Valley, at least the area south of the Delta, a couple hundred years ago was about half wetland and about half upland desert, and it's since been converted into basically uniform irrigated agriculture. And this is the upland desert part of that community, which still exists on the southern extent of the Diablo Range, which is where most of this story is going to take place. And it's home to a whole suite of unique organisms. The ones on the top are basically only found there, and then the ones at the bottom exist in some other areas, but still are fairly unique to the Southwest deserts. We have things like the San Joaquin kit fox, giant kangaroo rats, 3 other species of rare and endangered kangaroo rat, and then our lovely blunt-nosed leopard lizards, which are going to be the star of the rest of the show. So in case blunt-nosed leopard lizards are a new thing, I know even growing up as a herpetologist, I guess I wasn't a herpetologist as a kid. I was a budding, wanting to be a herpetologist as a kid. I basically never heard of these things because I didn't grow up in California. And I find even people that have grown up in their heart of their range have often never heard about them. So in case these are new to you, I wanted to go through a little bit of their background. They were actually on the inaugural endangered species list in the late 1960s. They're also one of California's fully protected species. They were listed out even before California or the federal government had an endangered species list. So we've known they've been in trouble for a long time. They're pretty short-lived in the wild, only 1 to 3 years, though we think in human care they could live easily 10 years. We have some that are about 7 years old at the zoo right now. They're pretty chunky lizards, at least for a North American lizard. They weigh 25 to 65 grams, which maybe doesn't sound like too much, but they're a big lizard. You notice them if you go out and see them. They're about 4 times the size of the bluebellies that are super common around here. They primarily eat arthropods, so lots of bug eaters, although they have some relatives that really like to eat other lizards. They're egg layers. They produce 1 to 3 clutches a year, almost always 4 eggs, 2 from each ovary. We see it over and over again because we ultrasound them out in the field. and they are very closely associated with rodent burrows. They really like to hang out around both ground squirrel burrows, but also kangaroo rat burrows, which in their ecosystem just sort of turns the ground into Swiss cheese, if you've ever been out in some of those San Joaquin Desert areas. So that's a little bit about who they are. And then another sort of key thing I wanted to walk you guys through before we get into the rest of the science is a little bit about sort of what their average year is and what their life is like. Because they spend more than half the year underground. At least that's what has historically been thought. They are laid as eggs in midsummer, like July-ish. They stay in the ground for a couple months usually, usually it's about 60 days that it takes them to incubate. They then hatch out. Over here, you get your little hatchlings that are going to be above ground for a month or two. They come out in late July, and they start to disappear by the end of September. You then generally don't see any lizards until the next spring. In the spring, say March to April, they come out. You see the yearlings, and the yearlings are already sexually mature. They're right on the edge of being sexually mature. They stay active through about the end of July, and then they become dormant again. They don't really overlap with the babies very much at all. If you go out in August, you'll see babies and you won't see adults for the most part. And then if they're really lucky, they go dormant, they survive again, they go down in like late July, and they'll survive to come back up again in April and do the whole thing again. The oldest one ever observed was 5. It was 1 at a site that had been monitored for decades. The vast majority don't make it past 2 years old, with over half of them only living that first year in the wild. So they're almost annuals in nature, at least in the relic habitat in that last 5% that we're able to study them in. And as a result of all this and all the habitat loss, they have been blinking out at an alarming rate, despite being on the endangered species list. So this plot over here shows the sort of 2 main genetic groups. Depending on your philosophy, you would call them 2 separate species. This northern group and this southern group last shared an ancestor about 2.5 million years ago, kind of on the order of us and chimpanzees, just to give you a sense of the timing. And the reason there was that big break, I think, was the historic Tulare Lake Basin, which would have separated these northern groups and the southern groups. The northern group in particular has really been declining rapidly. They've lost like 30% or more of known populations just this century. And the ones we work with are in the Panoche Hills region, which is on the eastern edge of the Diablo Range. And in 2019 and 2020, we watched the last population on protected land in this northern clade basically disappear. We thought it had completely blinked out. We saw a dozen animals out there at the start of the season. By the end of the season, it was down to 6. We confirmed this with fecal genotyping as well as our behavioral observations. We got emergency permission to collect 5 of them that year to the following year to bring in and try to get them to breed in captivity so we could start trying to rewild this area, learn how to rewild them, and possibly then apply that to the many other areas that they've recently blinked out. And we've been successful so far. This is our first little baby. I like to call him Smiley. And since then, we have actually produced 234 hatchlings. Thank you. And these are multiple generations of hatchlings, so we've had success getting them to breed in captivity, fully rearing them in captivity and breeding them. And we have begun doing all sorts of things about them. I'm getting ahead of myself. We are trying to not just make animals to release to the wild, but we want to produce wild type animals to release to the wild. And it's very easy for an animal to become almost instantly domesticated. It only takes a couple generations if you're not really careful that you end up propagating traits totally by accident that are beneficial for an organism in a little box in human care, but not beneficial to them in the wild. So we're measuring kind of everything we can measure non-destructively about these animals, measuring wild ones, and then trying to compare to make sure that we're producing animals like the wild ones, and then also so we can see which ones do well, and we can possibly target our rearing protocols to produce animals with the traits that are most successful in the wild. And just to give you a sense of that, I'm going to focus on this growth and maturation piece. But if anyone's interested about any of the other things, I could talk your ear off all day. So these are growth trajectories of males on the left, females on the right. Age is the x-axis. The y-axis for this is snout vent length, which is a sort of standard measure of the body length of a lizard. Size of maturity is shown with that dashed line going across. The gray shaded areas are when they would typically be dormant. The white areas are when they would typically be active. And then the dark line are measurements from wild animals. The orange line are measurements from our captive colony. And all the little skinny lines are the individual lizards that we've measured over time. And the main take-home thing is they look really similar. So our animals in human care are growing up, they're becoming mature at the exact same rate as the wild ones. So we think we're being able to recreate those natural wild type conditions in human care quite well, and we're getting a very similar phenotype from our babies. So then we don't want them just to live at the zoo. At about 10 months old, each spring, we release them. And we actually got to have a bunch of folks from Save Mount Diablo join us last year for our release. We've released 72 so far back to Pinochet Plateau. We are anticipating releasing over 50 next spring. Fingers crossed. We have 69 young ones that just went down for their winter sleep. And we put transmitters on each one of them. This is one of our collar type transmitters, so we can track them all throughout their lives to see how they're doing. Now, one thing that I haven't mentioned yet, but is worth pointing out, is that all this plant you see, or almost all of it, is an invasive grass called red brome. Almost all of it's red brome. The only things that aren't are these little bushes here. Those are native ephedra shrubs. And these animals did not evolve in a landscape dominated by grasses. The grasses also came within about the last couple hundred years. They evolved in a landscape that had small forbs, had bushes, a few bunch grasses, and then these nice little channels between everything that the lizards used as their highways and the runways. It wasn't just the lizards. The kangaroo rats did this too. The snakes did it. The antelope ground squirrels. The whole ecosystem evolved together without these carpeting grasses. And we've started to find that, especially for our blunt nose, and we think for a lot of species, these grasses cause a problem. You put a blunt nose in grass, it just sits there and doesn't move. We tried it. We documented it. It will stay there for months, basically, and not go anywhere. Where when you put them in an open area where they can see where they're going and they're not bumping into stuff, they run around, they live like a natural wild lizard. So this first year, this is from 2023, you may remember we had a record wet year after many years of drought. The grasses just went insane out there. It became a full carpeted mess. So we tried in year one to, oh, I didn't show it. Yeah. This area, you can barely tell, but we mowed this by hand. We mowed about 20 acres out there with weed whackers trying to take care of it before we released our lizards. But it didn't really help. We left enough thatch behind, we just weren't able to rake it all out. The lizards still didn't do very well. So now we've started doing some moderately large scale grazing trials out there. In this part of the San Joaquin Desert, the southern end of the Diablo Range, sheep farming, or sheep ranching, was sort of, it's like the historic economy of the place, at least historic for the Europeans that wound up there. And they're having some hard times, so we're sort of seeing it as this win-win where we can bolster historically relevant economic practice, the sheep raising, and use it to do some habitat management. So we're trying to figure out what is the grazing practice that will best reduce this cover in a way that is beneficial for the native plants and beneficial for the native animals, but knocks back the grass as much as possible. So we actually kept these sheep on for only 10 days in this plot and then moved them on. And still, if you go out there now, almost a year later, you can still see the signature where the sheep were grazing, and it cleared the area out pretty well. We're planning on keeping that going for a few years. And we have control plots and grazed plots that we're trying to compare and see what the lizards are choosing as well as we're monitoring the plant communities out there to see if we are actually encouraging the native plant community to be restored through this grazing practice. The other thing we're doing is we're really cranking up our monitoring efforts. So instead of using that hand telemetry that we've always used in the past, and we got to see with the pond turtles earlier today, we are converting to the modern era, and we are using these digital tags that are quite a bit smaller, and they talk to remote nodes that we put out on the environment. And each of these little dots, those nodes then talk to a central tower, which is part of the MODIS network, if you've heard of MODIS. And what it basically allows us to do is every few minutes locate where an animal is at pretty high precision, within tens of meters. And we can do it again, like I said, every few minutes. And it's really revolutionizing our understanding of what the animals are doing on this landscape. So for example, instead of getting a few dozen locations for an animal over the course of the season, we got over 300,000 pings for animals in the 2024 season alone. And we can sort of piece together a much more complex and detailed story of how the animals are using the landscape. So in this plot, the blue stars are where we've actually hand-tracked lizards to. And then all the other points are estimated from that node array. And this is just for a 13-day period, right after release. And so we can see a lot more details of the landscape use. We can basically see where the animal has moved, what habitat is dried out, what habitat it hasn't, in much greater detail. And it also allows us to answer questions like, what's home range size? When are the animals active? In greater detail. So this plot is showing another lizard, and the yellow line was how we would estimate its home range just from our hand tracking. And the light blue is how we would estimate their home range and their habitat use based on the node data. So we're getting a totally different answer to how they're using the habitat, really, when we're using the node data. It's also telling us more about when they're active. Remember, I told you at the beginning that there is this extended period that they're dormant. The adults from like late July, and they don't come up till April. And that is, it's not untrue. It's mostly true. The thing is, they're much jumpier that time of year. And so if you go out there as a big mammal looking for them, they dive into a hole and you don't see them. When you're not there and you just let them do their thing, it turns out they are coming up whenever the weather is nice. So this is showing the date on the x-axis. The y-axis is detected activity from that node array. And the red triangles indicate when we actually saw them. We went out there, we saw they were there. So a physical confirmation. And we can see that, yeah, they're active when we think they're active, but then during the whole period we think they're inactive, they're coming up opportunistically. And at least on a couple of those occasions, we went out and we saw them there. So we know it's really happening, which is giving us a really different understanding of their natural history on the environment, of how they're existing on the environment. All right, so I'm already out of time. I'm just going to give you a sort of quick summary. I said we released 35 last year, so how are they doing? 29 of them are still alive, as far as we know. They made it through the season. We will check on them in the spring. We're also seeing those pings come up when the weather's really nice. At least some of them are still coming up opportunistically. We have seen courtship of our releasees, although we have yet to document reproduction for many of our releasees. But still, early days. And we're going to keep this going until we get to a point where we have at least 50 females naturally born out there successfully reproduce. And we're going to see that as okay, we think they're at a point where they can build up to carrying capacity by themselves. We'll take the lessons learned and start applying them to other sites, places that organizations like Save Mount Diablo have purchased for restoration and need to have their wild birds back out there. But I'd like to thank you all. I had many generations of people sort of in classrooms helping with this work over the years. We've had multiple funders, notably here. Save Mount Diablo, through one of the Mary Bowerman grants, allowed us to purchase a scientific weather station out there that we've only had running for a year, but it's already transforming how we're maintaining our lizards in human care, especially over winter, because we know exactly what temperatures they would experience in their burrows over winter now instead of having to just guess. So thank you so much. If I have time, I'm happy to take questions.
Do your hybrid tags work when they're still underground?
Yes, they work for 3 days on battery power. And then they go quiet until the animal comes out again. And once it's in the sun for 10 to 30 seconds, they start pinging again. Yeah.
Okay. Hello everyone, my name is Prakrit, and today I'm going to talk a little bit about the ecology of scorpions in the Diablo Range. So the Diablo Range is actually a fantastic place to look for scorpions, and there's very little information available out there. So I hope to fill that gap and talk a little bit about how interesting these species are. But first, to get started, I want to talk about how important Save Mount Diablo and the Diablo Range has been to my introduction to scorpions. So like many people growing up in the Bay Area, I thought about scorpions as something that lives in the deserts and something that maybe is not as easily found so close to home. But at a BioBlitz by Save Mount Diablo in 2014, when I was about 10 years old, I saw this scorpion right here, which was found by Kenichi, who's in the audience today. And that was my first introduction to Bay Area scorpions, and it's really what got me excited. A few years later, I continued to look for scorpions in the Bay Area to try to learn more and more about them. And I started trying to do photography on my own. I'd like to think my photography has come a long way since here, but a lot of these early experiences in the Diablo Range were really instrumental to me getting started in working on these species. So now I'll start to talk about the scorpions of the Diablo Range. Now, as I mentioned, there's very little information about them out there, so I've tried to compile information on each of these species in sort of a field guide kind of format. So please feel free to take pictures of the slides, and I hope this information can be helpful for people who are interested in the biodiversity of the Diablo Range to learn more about our native scorpions. So here are the 10 scorpion species that have been confirmed from the Diablo Range thus far. They encompass several different families and a great range from very large species to very small species. This is actually an unusual amount of diversity, especially for an area with this kind of habitat, an area that's relatively far north compared to where the bulk of scorpion diversity is in North America. In fact, this is more scorpion biodiversity than is found in 44 US states. So this is really quite a remarkable area for these animals. So first I'll start talking about the western forest scorpion. This is a species that many of you are likely familiar with. It's one of the most commonly encountered scorpion species in the Diablo Range. And this is a species that can most often be found in habitats such as this. It lives in forests and woodlands. Many of you might have seen this species in places like Mount Diablo or places like Mount Hamilton where these pictures were taken. And it can be seen in these parts of the Diablo Range. And for a note on these maps, over here on the right, I have made a model of the distribution of this scorpion based on parameters of climate, soil, the topography of the land, and so on. So it kind of depicts what theoretically might be suitable for this species. And then over here on the left, I have a model of a map that I drew by hand based on records from iNaturalist, based on my own surveys, and based on museum records that suggest what the more accurate range of the species might actually be. But for a lot of species, the range of the species is actually not very well known, especially in the interior parts of the Diablo Range. So that's a big step that needs to be filled by more scientific surveying, and it'll really help us learn more about and conserve these animals. Next up, we have the sawfinger scorpion. This is another species that many people might have seen. This is a smaller scorpion and can be identified by its long, thin, and curved fingers. This species is really restricted to rocky habitats in the Diablo Range. It really likes outcrops of rock like what is shown over there, or even talus like what is shown over here. And this is a species that is found essentially throughout the Diablo Range, and all of the Diablo Range is really high-quality habitat for this species. Although strangely, it doesn't seem to be found in the Santa Cruz Mountains or East Bay Hills, even though the habitat seems theoretically suitable. Next up, we have the California common scorpion, or Pyroctonus sylvestri. This is a species that is also fairly common in a lot of the Diablo Range, but especially in the northern portions, and can be identified by its relatively dark coloration and the marbling on the carapace. And I actually have some examples of it if people would like to take a look. Pass them around. Yeah, so this is a species that in the Diablo Range is generally found in open habitats. It really likes sagebrush scrub. It also really likes these kind of open, more grassland-type habitats in the northern parts of the Diablo Range, and it can achieve really high density. In places like Altamont Pass, you can see dozens of them, even without using a UV light, just by walking around at night. And this is the distribution of this species in the Diablo Range, and I'll talk more about the distribution of this species and why it's a little bit more unusual with disjunct populations a little bit later in the presentation. Next up, we have this species, the Monterey woodland scorpion. This species doesn't actually have a common name, but I thought this was fitting. And this is a smaller species, although it looks superficially similar to the western forest scorpion. It has a more uniform color, and it has proportionally larger claws. It also has a little bit different habitat preferences. So this one lives more in woodland type habitats, but really can be found pretty much wherever it can find moisture. So even further south in the Diablo Range in desert type of habitats, you can find it as long as you can find these little dry creek corridors where there's a little bit of moisture. And you can see a little bit of its range over here. This is one of my favorite scorpions in the Diablo Range, and it's one of the first ones I learned how to identify in the Bay Area. But I would really suggest if you want to try to look for it in the Santa Cruz Mountains. It's much easier to find. This species right here is very interesting and very personally important to me. This is a devil scorpion in the genus Paravegevis, but it's currently unassigned to any species, and it most likely represents an undescribed species. And this was actually something that I found out about in 2019 while doing surveys near Panoche. And this is the first time I found an undescribed species of scorpion, and this is what really got me interested in working on the taxonomy of scorpions, and that's something that I've continued to this day. This species I have not yet described so far because it seems to have opened a taxonomic can of worms. This genus needs a huge revision, but it's something that I'm excited to work on in the future. This species can be found in desert-type habitats like what are shown over here, and it is most often found in the southern parts of the Diablo Range, although along the eastern margin it can be found all the way up to the Antioch area. Next up, we have another woodland scorpion, similar to the Monterey woodland scorpion, but this one is Gramlois evi. And this is one I've recently begun working on and found out that it's much more interesting than I had previously considered, because this actually potentially represents a complex of 5 or more species across California. An example of one from the East Bay Diablo Range region is over there on the right, but one from the Sierra Nevada is seen over here closer to me, and you can see the shape and size of the claws is pretty different between the two populations, and they're almost certainly different species. This one lives in woodland-type habitats and is really restricted to moist areas, and things like creek corridors can be good habitat for it. And in the Diablo Range, it's one of the most range-restricted species. It's only really found on Mount Diablo and adjacent parts of the East Bay Hills. Other populations in California are found further up the coast ranges, up into the Klamath Mountains, towards the Oregon border, and in the northern Sierra Nevada, but they almost certainly represent different species to the ones in this area. So this might be an endemic scorpion to the Bay Area, which would be really cool. Next up, we have the California swollen stinger scorpion. This is a very shiny species. It's a relatively large species, and it can be very easily identified by not only its shiny appearance, but also the swelling on the stinger in males. Hopefully that is visible right there. And this species is ecologically quite different from a lot of other California scorpions because it's an obligate burrower. And you can most often see it in its burrows with its claws sticking out of the hole, especially if you look at night with UV light. It often lives in chaparral-type habitats, like what is shown here, although sometimes it can be found in woodland as well. Pinnacles is really the only good place I've found to look for it in the Diablo Range area, but it is relatively broadly distributed in the southern part of the Diablo Range. And next up, we have the Superstition Mountain scorpion, this one right here. This one is my favorite species of scorpion in California. I think it's very unique and very interesting. It's part of a monotypic family, the only known monotypic family of scorpions globally. And it is very small, has a very unique pattern, and is also a habitat generalist. But in no habitat in the Diablo Range is it particularly common, except for one, which my surveys have found out recently. So typically this species is thought to be found in more desert or chaparral-type habitats, but throughout most chaparral in the Diablo Range it is absent, and in the Diablo Range the only place that surveys have shown that it is common is in this high elevation incense cedar habitat up on San Benito Mountain. And I have some more examples of this species as well. And this, yes, you can see the distribution of this species in the Diablo Range. And this is particularly interesting because this species, the model of the habitat suggests that it should be present in the Mount Hamilton Range. And it actually doesn't show these southern parts as being all that suitable, which potentially suggests why they're not very common in that area. So additional surveys might actually expand its range further to the north. Another thing about this species, like some of the other species I've talked about, is that this might represent a species complex, which might help to understand why we don't fully comprehend its range right now. And next up, we have the dark hairy scorpion. This is by far the largest species of scorpion in the Diablo Range. And the species is also one of the more range-restricted species in the Diablo Range. Really, this is a San Joaquin desert scorpion, but it barely ranges into the Diablo Range in the Panoche area and in the Coalinga area. It really can only live in these very open habitats, and especially it likes the flatter open habitats more than the steeper open habitats in my experience. And here's an example of its range within the Diablo Range. And I have an example of this one as well. Now, with that out of the way, I'm going to get to Pareoctonus variabilis, which is the species that I have been studying for the Mary Bowerman grant, and one of the most interesting species in the Diablo Range. This species can be very variable, as its name suggests. It can come in this light form like this. It can have a dark form such as this one and a more orangey form like this one. And it can be found in quite a few different habitats throughout the Diablo Range. I do have some examples, of course, of this species as well. And to talk about its habitat a little bit more, so the habitat of Pareoctonus variabilis can be very variable, again, as the name suggests, throughout the Diablo Range. The most typical habitat for the species and the most abundant habitat is typically habitats such as this. It's kind of a desert type of habitat, steep slopes with a lot of fine soil and rocky habitat. They really enjoy burrowing in fine soil, although they're not obligate burrowers like the Anuaroctonus. This is a species that's almost always found in association with burrows, especially in this kind of habitat. It can be also found in areas such as this. These really steep soil cuts are great habitat for this species. But it can also be found in more atypical habitat. This is kind of like a more flat and rolling hills type habitat where they can also be found, but in small populations. They can also be found in the incense cedar habitat up on top of San Benito Mountain, which is really unusual habitat for them. It's really the only forested habitat that they can be found in. They're found in more chaparral types habitats lower down on San Benito Mountain. And they can be especially found in chamise chaparral type habitats further north in the Diablo Range. So almost all of the disjunct populations of this species further north are associated with chamise and other similar chaparral plants. And lastly, in the San Joaquin Desert, the species can be found in completely flat habitats such as this, although my preliminary work is suggesting that these are probably a separate species. Now, the range of this scorpion can be seen here, and as you can see, the entirety of the Diablo Range does have a theoretically suitable climate for this species, but it is absent from much of the northern Diablo Range. And my hypothesis is that this comes down to competition with Pareoctonus sylvestri, which is a closely related and morphologically relatively similar scorpion species. So this Pareoctonus variabilis is pretty varied across this range, as I was mentioning, and you can especially notice that lighter phenotypes are found here down near the south, and darker phenotypes are found up here near the north. And these darker phenotypes that are found in core Pareoctonus sylvestri habitat are morphologically more similar to Pareoctonus sylvestri than these lighter phenotypes, which once again suggests to me that there is some kind of ecological factor which is suggesting that Pareoctonus sylvestri is a better adapted scorpion to these northern areas over here. This is the distribution of the two species overlaid upon one another, and this now makes the distributions make quite a bit more sense. You have Pareoctonus sylvestri up here further towards the northwest, and Pareoctonus variabilis down here towards the southeast. So this kind of shows like a more desert versus like a more temperate and chaparral kind of habitat split. And these species mostly have an allopatric distribution. The distribution is parapatric in some areas, which can be highlighted in orange here. And most interestingly, they do have certain sympatric populations, which are shown here in red. These I find particularly interesting, and I'm going to zoom in on a couple of them right now, because they follow two completely different trends. So this sympatric population right here is found in the central part of the Mount Hamilton range, where Pareoctonus variabilis is almost completely associated with small patches of chamise chaparral, while Pareoctonus sylvestri is broadly distributed. It's found in lowlands over here in a more desert kind of habitat. It's found in sagebrush chaparral over here, and it's found pretty much just everywhere. This population right here at Mission Peak is also very strange. Pareoctonus variabilis seems to be restricted to only a couple hundred meters along a particular sandstone outcrop. This habitat is the complete opposite. This is down south near Panoche. And now, instead of Pareoctonus sylvestri being widespread, Pareoctonus variabilis is widespread, again found in lowland desert type habitats, in sagebrush chaparral type habitats, in chamise chaparral habitats, in just basically wherever it can live. And Pareoctonus sylvestri is now the one that's restricted to a very small area, right up here near the top of San Benito Mountain. So currently, I have no idea what's going on here, and I would love to hear anyone's input on why these species have such a bizarre habitat preference. Now, finally, to wrap up, I'll talk about the next research steps in Pariocnus variabilis and what I'm working on with this grant. So I've been especially looking at these different population segments of Pariocnus variabilis. The colors here point to different genetic clusters within the species that potentially might be species-level splits. But their ranges seem to overlap, and I think that additional genetic work with next-generation sequencing is needed to really resolve the details of this, and this is what I've been working on with the Mary Bowerman grant. But in addition to that, looking at the variation across different localities and competition with Pariocnus sylvestri, or potentially even gene flow with Pariocnus sylvestri, is something I'm very interested in the future. So with that, I would like to thank everyone for listening. I would especially like to thank the individuals here on the screen who have helped me with many aspects of this project. And I'd like to open this up to questions. And this is a Pariocnus variabilis cannibalizing a small Pariocnus variabilis near Panoche.
That was a very interesting talk. Can you help us a little by relating the anatomy that you're distinguishing these by for their survival in their environments?
Yeah, so a lot of the details of scorpion anatomy are pretty closely linked to either substrate or to diet. So one thing in particular here, you can see the size of the claws. The size of the claws for most species is related to the musculature of the claws. And species that rely more on their claws, such as burrowing species, since they can't get their tail out of their burrow to sting their prey, they typically have broader and stronger claws, versus species that hunt more out in the open and more use their venom to subdue prey. Another thing you can look at is the legs. So the legs of the scorpion are more robust in burrowing species, of course, because they use them to burrow. But also the hairs on the legs are another key factor. Species that burrow in sand actually have long, thin legs, unlike those that burrow in clay. And they use the hairs on their legs to help them burrow, while those in clay soil end up having clay get stuck on their legs, so they don't have many hairs on their legs, and they instead use strong, robust legs to burrow. So those are some of the features that relate to the ecology.
Could you tell us a little bit about how you find these, notably using the black lighting and the flipping? I understand the black light, but I don't understand the flipping.
So the flipping is just turning over surface objects, such as logs and rocks, and the scorpions can oftentimes be found underneath. Some species of scorpion, that are more living underground, such as the woodland scorpions, can be difficult to find by blacklight because they're not always active on the surface. So I find a more reliable way to find some of those species is by turning over surface objects.
Quick question. Some of those scorpions are alive in the containers, yes?
Yes.
Okay.
Good question. Yeah, one, two.
You've done a tremendous amount of work finding this massive distribution, unless you've got a lot of help. So can you reflect on numbers of individuals that are out there?
I see. So scorpions in the Diablo Range can sometimes reach very high densities in some of these locations. Species, especially like the sawfinger scorpion, you can have certain areas where you can look around with a black light and have around 30 individuals in front of you. Other species are much more difficult to find. In all my surveys in the Diablo Range, I've seen the dark hairy scorpion only once within the Diablo Range, although it's a little bit more common further south. But I think right now there's a lot of work to be done on the actual population density of the scorpions. Really only one set of studies has been done in California on dune scorpions in Southern California that looks more at population density kind of questions. So I think that's one place where there's a lot of work left to do, and I think it has important implications for things like conservation of these scorpions.
Thank you, Prakrit. Yeah, if any of you have more questions for him, which I'm sure you do, let's wait till lunchtime, but thank you for that. That was excellent. Thank you.
Hi, thank you all for joining today. My name is Jacob Woodall. I am a master's student at San Francisco State University in the Amphibian Conservation Lab working under Dr. Vance Friedenberg, and I'm going to be presenting my research today for my master's project. So the disease we'll be discussing today is called chytrid, or amphibian fungal disease, clinically known as chytridiomycosis. It is caused by the fungus Batrochytrium dendrophytidis, or fortunately also referred to as BD, which is much simpler to say. BD is an aquatic fungus, so the zoospores, which is this, the zoospores, which is this stage, is the mobile stage and can be transported aquatically or through skin-to-skin contact. BD feeds on the skin of amphibians and ultimately can cause mortality due to preventing electrolyte transport across the skin. It has been linked to declines and extinctions globally in North America, Central America, South America, Europe, Australia, and in California, it's been linked to the local extirpations of several frog species in the Sierra Nevada region. The species I'll be discussing today is my personal favorite frog species in California. It is the western spadefoot. They are Spia himandi. It is a California endemic species being found in the Great Valley down the south central coast. Recent work has identified two distinct clades in the species. There's a northern clade and a southern clade that's divided by the transverse range. It has been recently proposed to be listed as threatened under the Endangered Species Act and has been petitioned by the Center for Biological Diversity to be listed as threatened under the California Endangered Species Act. So hopefully there'll be more protections for it soon. It also is a species of special concern in the state. It is understudied, particularly the adult life stage or postmetamorphic life stages. Much of the previous work in the species has focused on the larval life stage. This family Scapiopodidae is unique in that they have the shortest larval stage of any continental North American amphibian. The western spadefoot can metamorphose in 35 days from egg to froglet, which is extremely fast for a species in our area. And following in this understudied portion, much of the previous work for the species has also been concentrated in Southern California. Prior BD sampling in the species has been done by the USGS, again in the southern clade, and they have found infection prevalences of 0 to 31% at sites. At sites that are BD positive, they have found prevalences from 7 to 31%. And prevalence is just the percent of infected individuals in the population or in the sample. And here's a range map for the species to kind of orient you about where we're talking about. This red line is my approximation of the transverse range, and the gold star is my study site that, where I've conducted all my research that I'll be discussing more. And it is Carnegie State Vehicular Recreation Area, which many people are probably familiar with. It is a off-highway vehicle recreation area managed by the California State Parks. It has a very, very rich history, both in the park and in the Crow Hollow Valley, both naturally and anthropologically. It's the intersection between the Diablo Range and the San Joaquin Desert. And there's been many, many, many generations of herpers to cruise this road looking for all sorts of amphibians and snakes in the spring. Prior studies by the park scientists at the site have looked at BD and California red-legged frogs, the movement of western spadefoots, and at spadefoot larval morph plasticity. The BD study in the park was part of a larger genetic population study of California red-legged frogs in Central California. They tested 18 red-legged frogs in the park and found 12 of them positive for BD. Their infection intensity, we report that for BD as a zoospore equivalent, so the approximate amount of zoospores that are on the frog based on the DNA fragments that we're testing for. And they found ranges from 2 zoospores up to 4,200 zoospores. And generally, for other species that we've measured in, we start to see mortality in certain other species at about the 10,000 zoospore level. So for my project, I was interested in investigating the BD infection prevalence and intensity in the northern population of the western spadefoot. There's a historical component to my project in which I went and I swabbed all the available museum specimens, looking at the post-metamorphic life stages, so the froglets, juveniles, and adults, and then a contemporary component where I am looking at prevalence and infection intensity, looking for differences between life stages for metamorphs, juveniles, and adults. Previous work in our lab for other species has shown that metamorphs generally have the highest infection prevalence and infection intensities. And I was also interested in if there's any seasonal trends. Spadefoots are a very terrestrial activity limited species. So they have a very defined terrestrial activity peak in the spring, and then they'll head down into the burrows where they may or may not emerge depending on climatic conditions until the winter. However, as many master's students and PhD students will tell you, projects never go to plan. Unfortunately, last year, it was a very poor rain year at my site, and we had no successful metamorphosis. We did see successful reproduction, but the pools that the spadefoots reproduced in dried out before they could metamorphose out. So I already kind of lost some of the life stage data, and there are only juveniles and adults present, but we still had the seasonal trends to look at. And so here's a picture of the two of the western spadefoots from our site. On the left, we have a juvenile frog, and on the right, we have an adult frog. Previous work looking at a histological study that looked at the gonad development in the species had found that adults are generally 37 millimeters or greater in snout urocell length, or snout vent length. For our sampling, the sampling for the museum specimens and the living frogs is the same. We swab them on their ventral side, doing 10 swipes on the stomach, 5 swipes on the inside of each thigh, and then 5 swipes on the back feet. We target these areas because these are the areas that are most likely to come in contact with the other frog species if they're amplexing each other, and they're also the areas that are most in contact with water when they're active in breeding. And it's where we typically are finding the infections. I am a bit of a busybody, so I also ran several side projects at our site because I couldn't help myself. To learn when we should be targeting these surveys, we installed some passive autonomous recording units to conduct passive acoustic monitoring to figure out when they were starting to breed. I also did a separate study where I conducted time series surveys to determine when we should be surveying in a given night for a frog. So these were overnight surveys where I went out once a month and surveyed 3 times spaced evenly across the night, starting an hour after sunset, which were very exhausting but very illuminating. Because we were collecting morphometric data to help us identify the adults, and it's just standard data to collect as we're handling the individuals, I've also, using a paper, developed a scaled mass index for the species to look at body condition across time, like what trends we might be seeing. And then because we're doing these standardized surveys, I've also developed an environmental covariate of terrestrial activity model to see when we should be doing these surveys for the species, especially going forward, if they get listed, when people should be looking for adults. Most of the presence-absence surveys for the species focuses on the larval stage. Larvae are very contained, obviously, to water bodies. They're not going to be dispersed through the landscape. However, as we found out at our site and other sites, they don't breed every year. So if you are trying to do these targeted larval surveys and it's a non-breeding year, you might miss the presence of the species. And this is, sorry, I should have added. This is our audio moth recording unit, the autonomous recording unit that we were using for the acoustic studies, up-close version, and then here is it deployed in the field. My coworker who does this a lot with birds advised on the weather protection for it, and we found that this double-bagged method with desiccants is both cheap and works great. For my historic methods, I visited 2 local institutions, the Museum of Vertebrate Zoology at Berkeley and the California Academy of Sciences. Fortunately for me, the vast majority of the post-metamorphic northern clade western spadefoots in collections are at these institutions, which was a breeze then to sample. And we took the frogs out of jars and swabbed them as we would for the live frogs. And here is my team of undergraduate researchers diligently at work swabbing at the Museum of Vertebrate Zoology. This project would not have been possible without their help. For my contemporary study, we conducted nighttime visual encounter surveys, which is a standard amphibian survey approach where you just go out with a flashlight held at eye level and the frog's eyes will reflect back the light to you, so it makes it easy to find them. We recorded environmental conditions at the start of surveys, captured frogs in containers to avoid potential contamination, and then set up an in situ processing station where we swabbed them and recorded the morphometric data, the physical data. So the snout urocell length, gape width, which is the widest part of the mouth, and their weight. And here's an example of, again, the sampling and those measurements. The gape width was a sort of QA/QC measurement. It's highly positively correlated with the snout urocell length. So by taking both measurements, if something was recorded wrong with weight or with one of them, it was easy to identify in the data entry step. And here's our team. Our in situ processing station was the back of my car, which worked wonderfully. Back in the lab, we extracted the DNA from these swabs, and we ran a quantitative PCR to determine the presence of disease and to determine the amount of the zoospores present. And it's just a really fancy way of saying we are targeting a certain region on the chytrid fungus that is highly conserved and isn't found on other fungus species. And when we find that region, our samples, it attaches a dye to it. So the machine reads the amount of fluorescence from that dye to determine the how much of it is present. Unfortunately, the results were great for the frogs, but devastating for me personally. I've tested 77% of the historic swabs so far, and all of them have tested negative for the disease. Fantastic for them. There are two terrible type of results to get in the laboratory setting, all positives and all negatives. With both of them, you then have to do a lot more work to ensure that your results are accurate. And so for ours, we run positive and negative controls on all of our plates to help to let us know if we have contamination or if our reagents are acting appropriately. And then, so this shows the number of frogs present in collections from each of the counties. There's a major skew in collections for the species, largely found in the San Benito, San Joaquin, and Monterey, or Madera, sorry. And then this is the same data, but looking at the decades that the specimens were collected. Prior work in our lab has found that chytrid in this area generally starts to show up in the 1960s and will rise in infection prevalence through like the 70s and the 80s. Unfortunately for me, or unfortunately for frogs, they just had 0 infections. And here is the results from the contemporary swabbing. Again, all these frogs at our site tested negative for the disease, which is, again, wonderful for them. To check on these results, we also swabbed western toads, Syrian tree frogs, and California newts present at the site with the spadefoots, and they all also tested negative for the disease. The previous, yeah, there's something going on there. The previous work, again, with the red-legged frogs, was focused on sediment basins where the frogs are found. The water from those sediment basins flows into Crowell Hollow Creek, where we conducted our studies. So in theory, and the red-legged frogs are also found in the creek. We're not sure what's going on. And talking about the seasonality of the terrestrial activity, you can see here, most of our frogs are found in spring. We did survey through this time period. We just had no frogs. And again, here the results, 100% of the contemporary sampling has been run. Over 266 swabs were taken from spadefoots, all negative. 167 swabs were taken from museum specimens, all negative so far with 78% tested. Which, yeah, and this is me after doing plate after plate after plate. This is one of our favorite frogs from the collections. Now for the other studies. For the acoustics, we ran into an error with the recording schedule. We we set the timer for the recorder on the wrong time zone. So they were recording earlier than we intended. And as such, we didn't really get great data from 3 of them. We tried to target our recording placement at areas where the frogs had been documented breeding the most in the park. Again, we did not see very much reproduction in the park, and the areas where we did see the reproduction was actually areas that they historically have had the lowest, the least amount of breeding activity. So we sort of missed all of our windows, and of the 2 remaining recorders that were in the proper area, they had chorus frog leks set up underneath them, which are very loud and work to drown out most of the other sounds. And then otherwise, Tesla Road, Crowell Hollow Road is very, very active at all times of the night. And so the vehicle noise also made the data processing a little more difficult too. And here's one of our culprits. This is a Syrian chorus frog or Pacific chorus frog calling from the creek habitat. Now for the time series surveys. The first survey in January did not detect any frogs. It was too cold on that night. But the following surveys were successful, and I analyzed the results with just a simple ANOVA to look at if there's any differences in trials or month. Trials was not significant. So trials, again, are the 3 surveys spaced over the course of a night, starting an hour after sunset, and then, depending on how long the night was, every 4 to 3 hours, or 3 to 4 hours after. Month, however, was significant with the highest number of frogs, which matched our data observed in March. And so here's that data. It seems like it would be significant in these surveys, but it ended up not being so. And this was great because prior work with their currents has not looked at when they're active at night. And so there was kind of a lot of rumors present about when we should be doing the surveys and if frogs were most active like an hour after sunset, if you could go out 4 hours after sunset. And so the time series showed that if they're going to be terrestrially active that night, you can kind of go out whenever. I think my latest survey was at 3 or 4 a.m. and there's frogs present. For the scaled mass index, I followed the methods outlined in this wonderful paper by Pieg and Green in 2009. Previous body condition indexes just generally look at body mass divided by like length or snout urostyle length for our case. They had I'm not going to get super into the arithmetic behind it, but they had a different way to kind of create a scale for the different size classes that you might see. And it worked great with the data we were already collecting at the site. We found, which was super interesting, there's clear clustering between the life stages, with adult spadefoots again being greater than 37 millimeters snout urostyle length. Clear clustering in these scores between the juveniles and adults. There were seasonal trends present, and we had 3 frogs that scored greater than 20 on the index. All were females in late March and early April, when we would expect them to be potentially provisioning eggs, which is really interesting. And maybe this is something we're hoping to get more repetitions with and see if that's something we can use to identify females that might be doing that. And here is a graph of that result. So in the red are the adult frogs and the green are the juvenile frogs. On the x-axis is the score on the index, running from 0 to 30, and on the y-axis is the frequency of that score for our data set. And so there's just very clear clustering of the data, and we're not really sure why we're not seeing any like in between. It might be due to when we're seeing the juveniles and adults, there's that period where we're not really finding them. Sorry, I think I'm running out of time. For the juveniles, we saw a clear increase in body condition over the course of the active period from February to May. For the adults, they were pretty similar in condition throughout the season. This is not using a linear model. This is using a low-S model, which looks more at the trends present in the data and isn't really good for predicting out. There is this bump, though, in female condition present in that, again, that late March, early May period. For the environmental covariate study, it's a lot to go on here. I used a general additive model, which is better used for biological data. I had a lot of environmental predictors to start. Ultimately, starting air temperature is the only significant predictor with a nonlinear relationship. So there's kind of a peak of abundance with the temperature, and they prefer between 7 and 20 Celsius or 45 and 65 Fahrenheit. And days since last rain ended up being a non-significant, slightly negative association. And here is one of our emerging frogs. Again, thank you very much to Save Mount Diablo Foundation for the funding for this study, the ARC's Northern California chapter also for scholarship funding, and for Stillwater Sciences, a local environmental consulting firm for lending their autonomous recording units. Thank you to all these wonderful, wonderful individuals for their assistance with the study, including my committee members, our director for our genomics facility at State, my wonderful, wonderful undergraduate researchers. This study would not have been possible without their support and help in the field and in the laboratory setting. Elizabeth Gutberlet and all the wonderful Carnegie Park staff and Diablo Range district staff who made this project possible, and the several other wonderful individuals who've offered assistance, a lot of assistance with the project. And for all my field volunteers, I've had a lot of field volunteers with this project. It's a lot of data to collect. I've had close to 100 individual volunteers come out across different nights as we were surveying weekly through that period, which I forgot to add. Thank you.
Just for time, if you have questions for Jacob, please leave them for lunchtime. Thank you, Jacob.
My presentation is on nest site selection in northwestern pond turtles, Actinemes marmorata. You guys heard a background earlier from Dave, and I'm just gonna repeat information. Their historical range spans across western North America, so from British Columbia, Canada, down to Baja, California, Mexico. Their conservation status varies within the United States. In Washington, they are endangered. In Oregon, they are threatened. And in California, they are a species of special concern, but actively under review to become federally listed. They are California's only native freshwater turtle. There are two subspecies, the northwestern pond turtle and the southwestern pond turtle, and they are a temperature-dependent sex determination species, specifically type 1A. So when eggs are incubated in cooler temperatures, it results in males, and when eggs are incubated in warmer temperatures, it results in females. They face a variety of threats, including habitat fragmentation and urbanization, invasive species like red-eared sliders and bullfrogs, and then climate change, such as droughts that can lead to mass die-outs and reduced reproduction. So what is nest site selection? Nest site selection is the non-random placement of nests or eggs by a female turtle to maximize her chances of nesting and successfully produce offspring. The aim is that optimal nest site selection minimizes the mortality of females and maximizes their reproductive success. It has not been proven in western pond turtles, but the literature seems to suggest that they might have preferences. Nest site selection is considered an environmentally flexible response. It can impact a female's survival and their nesting success. It is a critical behavior in turtles because once those eggs are laid, there is no parental care. So the only chance a female has at passing down her genes is by selecting a nest site that leads to the success and survival of her offspring. It's considered a mostly plastic response with likely low heritability, meaning it's based on environmental cues and individual experiences rather than on genetics. So with it being a flexible response, females can adapt and respond to short-term conditions, but it doesn't easily evolve over generations. So a female turtle that's able to select a good nest site, that doesn't mean that her offspring will inherit that same ability. And then lastly, it can be constrained by inherited nest site fidelity. So females may stay loyal to a site that might not be as successful as other sites due to this nest site fidelity. Nest site selection is important because where they nest is going to define those moisture and thermal conditions that are going to define offspring sex ratios, their survival, and their overall fitness, which is their performance, so their locomotion, ability to swim, their morphology, so their size and limb proportions, their physiology, so their metabolic rate, their growth rate, their behavior, so boldness and dispersal. It's going to reflect the ecological pressures in the habitat, which is going to be influenced by both biotic and abiotic factors. So that's gonna influence the risk of depredation and inundation on the nests, the chances that the hatchlings reach the water after emerging, as well as the survival of the nesting females. So selecting the wrong nest site can have detrimental consequences to both parent and offspring. Next, I want to introduce you to my study. My field site is in Livermore, California. It is a 5,000-acre cow-calf operation that's grazed year-round. It's owned by Zone 7, the water agency, but leased by the Banke family. It is a spring-fed perennial stock pond. It is a silty clay loam soil that is there that directly surrounds the pond, and it is steep terrain. There are 45 to 75% slopes. And there have been 0 western pond turtle studies at this site until we began our research. But the pond is amazing and full of life. We have a lot of large red-legged frogs. We have killdeer that nest every year. We've seen the successful recruitment of the western pond turtle, so we see the hatchlings. We have no red-eared sliders, and we have no bullfrogs. Now I'm going to take you through my June 2024 and 2025 nesting season. So I'm going to break down this very colorful map that you guys are seeing. So in 2024, we had 20 successful nests, which are in green. In that same year, we had 11 failed nests, which are in red. And we also had a double clutch that year. So our first female to nest was also our last female to nest. She nested on June 6th and then again on June 26th. In 2025, in blue, we have 23 successful nests, and that year, or this year, we had 6 failed nests in orange. So in total, I had 43 successful nests and 17 failed nests. I collected nest site characteristics for all of these. In 2025, I introduced soil sampling for all of those successful and failed nests. In order to properly study and understand nest site selection, I deployed 50 random sites to sample. And so I collected nest site characteristics at all of these sites, including soil samples, in order to compare the sites that they're not nesting at to the sites that they are nesting at. So what does the literature say? Most published studies have been limited to descriptive accounts, and so it suggests that females prefer to nest on well-drained clay-sit soils with a slope of less than 15 degrees. For my preliminary data, on average, I found that our aspect was northeast facing, so about a 72 degrees. Our slope was 22.99, so well above that 15 degrees, but remember, this is very steep terrain. Distance from pond was 49.87 meters. The furthest nest was 161.5 meters away. However, western pond turtles are always nesting within a water source. So distance from water was 23.76. So that's whether they're nesting near the pond or those 3 creeks that connect to the pond that are also available to them. So distance from pond is just going to reflect the female's movement, while distance from water is going to reflect the habitat that the hatchlings are going to utilize once they emerge. And then lastly, our distance from canopy cover was 14.58 meters. For my preliminary data, when comparing successful nests to random nests, I found that western pond turtles select sites non-randomly, choosing distance from water and distance from canopy cover, and in soils with specific moisture and bulk density. So as you can see on the photos on the bottom, the image on the left is the north-facing hill, where a majority of the nests were laid, and you can see that there is a variety of canopy cover there. Where if you look at the image on the right, that is the south-facing aspect, and there's very limited canopy cover, and there are no nests that occurred there. So this is fantastic, because now we can actually prove that nest site selection is occurring in western pond turtles and what habitat characteristics they are selecting for. I do want to point out that at our field site, slope and aspect did not matter. Diving deeper, I found that when comparing successful nests and failed nests directly, only soil moisture and bulk density differed, indicating that that soil microhabitat is what predicts nesting success. So that soil moisture and bulk density is going to depend on whether the females are able to excavate and lay their eggs, or if they end up digging for hours and then bailing because that soil is just too compact for them to dig in. I also found that at a population level, the females altered their nest site placement between 24 and 2025, and that canopy selection was actually driven by low temperatures for the successful nests. This could be due to a cool temperature risk. So we know that the females leave in the evening to nest, they stay on the nest overnight, and then return to the pond the next morning. So they could be assessing that microclimate to see how that might impact their behavior overnight. And we know that there's limits to embryo development and their sex ratio, so cooler temperatures result in males, so maybe they're assessing that microclimate to ensure that those clutches aren't going to be male-dominated, because you need females in a population to survive, especially as a long-lived species. So these results show that all females may shift their nest site behaviors across years, but only the successful nests reveal a consistent thermally guided habitat selection, which makes sense because they are a temperature-dependent sex determination species. And then lastly, I found that western pond turtles that nest further from water nest closer to canopy. So in 2024, they nested closer to the pond and further from canopy, but in 2025, they nested further from the pond and closer to the canopy. So overall, it appears that canopy is playing an important role in nest site selection, likely as a thermal buffer. Western pond turtles are a plastic species, so it can lead to variation in their behavior, and this could be due to a variety of reasons, such as environmental conditions. 2024 was a lot hotter, and it received more rain that year. 2025, our nesting season was freezing. It was really cold this year, and it's been a lot drier. Cattle pressure presence could have changed. In 2024, it seemed like the cattle were there more often. Again, it's probably because it was hotter, so they were trying to utilize the pond more often as a resource. I do want to point out, so there's no questions on it either, I have not seen turtles trampled by cattle, and I have not seen nests trampled by cattle either. Predator pressure can also be an influence. However, this doesn't feel likely at this field site because we had 0 nests depredated both years. And then lastly, human disturbance plays a role. So 2024 was their first exposure to us. We were tracking them, manhandling them. 2025, they were getting used to us. We did notice an overall shift in their behavior, and it's hard to say whether that's on us or other conditions or everything put together. But in 2025, there was a lot more social facilitation that was happening. I noticed that on average, they were leaving in groups of 3 to 4 from the pond. They would go up to the pond edge together. They would look at each other. One female would decide to leave, and then the rest would immediately follow her. And most of the time, they were nesting within a meter of each other, which was pretty interesting. Did not happen in 2024. We do have 2 new grad students. We have other current projects going on. We have temperature-dependent sex determination happening. So we put temperature loggers in the nests this past summer. We put them in 15 nests. When we went to pull them, all the turtles had hatched, and so when we took off the nest plug, a hatchling went running, and we didn't want to disturb them because they overwinter, so we just popped the nest plug back on, and we'll be pulling the loggers in the spring. And then we're also looking at nest site fidelity. But all of this to say is that this research is important because it fills the knowledge gaps on nesting behavior in western pond turtles, which is an underrepresented species in conservation and in research. It allows us to examine site-specific differences across habitats, which allows us to gain insights into their ecological flexibility and adaptability. It can directly support local land managers by helping them make informed conservation decisions. And overall, it's promoting collaboration between researchers and ranchers. What's important is that protecting turtles means protecting entire ecosystems. Nest site selection is a very critical part of reproductive success, but has been historically under-researched. A 2023 review paper on nest site selection in freshwater turtles went through hundreds of turtle papers and only found 35 on nest site selection. And most of those 35 papers were on sea turtles and not freshwater turtles. So setting this up was like a work in progress, trying to go through the very limited research. But this not only improves our understanding and our knowledge on western pond turtles, but also contributes to the greater efforts of freshwater turtle conservation. I cannot do this project without so many people. Farley and Jackie from the NRCS and the ACRD were huge components to this project, and my advisor. A big thank you to the Binky family for allowing us onto the property. Of course, thank you to Save Mount Diablo. I would not have a thesis without you guys. But overall, so many people that worked on this project made it happen. Thank you.
Thank you, Maddie. Any questions?
Just a simple question. How do these turtles get introduced into these ponds?
Well, turtles can actually disperse very far, and their range spans across all of Western North America, and they used to be a lot more prevalent as species. So we've killed off the population by millions through turtle soup just within the Bay Area. But these ponds connect to greater water ecosystem. So we've had turtles that have completely left these ponds. They went up a creek, over a ravine, entered a reservoir, and then went up another creek. So they've traveled more than 5 miles. Yeah, my advisor likes to call them all-terrain vehicles.
Another question.
What's the distribution by sex in the population in your study area? And also, can males impregnate more than one female? Have you been able to determine that?
So our sex ratios are pretty even. Our population sits around 50-60 individuals, and it's pretty split in half between males and females. And then what was the second part? Yes, yeah. So I don't know how often that's been looked at, and if there's a dominating male that more likely reproduces than other males. It's more likely we do have some really large males compared to the other males. So it's likely that they might be the ones that are more successful in reproducing.
I was curious, you had mentioned about the overwintering of the juveniles, and could you expand a little bit more about the life cycle of these turtles?
Yeah, so females tend to nest during June. They can go into July. It's going to depend on both the environment and warmer temperatures. So warmer temperatures are going to speed up their gonadal cycles, but they also have to have enough nutrition and resources to allocate to that. So that's going to dictate nest timing. And then when they nest, they incubate for 90 to 120 days. In the northern population, the hatchlings stay in the nest, most likely to avoid the more extreme weather that we exhibit here compared to the southwestern pond turtle. The southwestern pond turtle doesn't overwinter, so those hatchlings just emerge. And then those hatchlings will emerge come spring, so most likely around April time when we start getting warmer temperatures again.
Yeah, I was curious about the social facilitation that you mentioned. Do you have data on who was leading those female groups that you mentioned and versus who is following? Are there any characteristics that determine that?
It's really hard to tell who's who leaving the pond. What we could do, and we've done it for hatchlings, is you color code them with a marker. So we could do a color code on their shell, so when we can see through the binoculars who's leaving first, and that's not a bad idea. But no, I haven't done that. I do when they leave at staggered times, they pick a general direction to go in, and I kind of note the direction, and then when I go to check on them in my head, I can figure out for the most part who was who, based on the direction and where I find them. But that is a smarter idea.
Any other questions?
I'm curious about, we've been trying to document them on some of our local stream systems, and we've had success with photographing a few. And do they nest on running water stream systems, or do they need an actual pond to nest there?
No, they won't need a pond. If you're looking at it like a creek setting, they'll just move upland from the creek to find habitat to nest in. So it's not like they need a giant water source like a pond. Yeah.
But they would have similar travel distances, you would expect?
Yeah, it also depends if Or is it like an urban creek?
Urban Creek, but it's a very large open space area
Okay.
within an urban area.
Okay, then probably they'll stay within like the confines of that. You get into the issue of more urban settings with reducing their habitat, so then that's when they go and venture, and then now they're crossing roads and going into neighborhoods and trying to find more suitable habitat, which is also maybe playing a role in the distances that they're traveling. But if you have available suitable habitat, it's likely that they're staying within that area.
Thank you again.
It's a pleasure as a Stanford alum to come to the Berkeley campus. How about that big game? Broke our 4-game losing streak. I'm going to talk about animals greater than 6 inches tall, which makes this different from the rest of the presentation. But I'm a physician scientist. I started the Institute for Historical Ecology. We study California's historical flora and fauna and their distribution, much of which was poorly understood, to guide restoration and rewilding. I'm pleased to see Save Mount Diablo as restore in the middle of their byline, because a lot of land trusts conserve lands, which is an important first step, but I think need to focus on rewilding. There are 2 native mammals in California that help mitigate wildfires, elk and beaver. Beaver because their streams and ponds may provide barriers to fires and provide water sources and for firefighters actually, and elk because they mow the grass. On the right, I've published 4 papers on elk in California, largely calling for their recovery and restoration, and talking about how they're often blocked by freeways from recolonizing the rest of the state. They reduce fire fuel loads. This is why I would argue elk are especially important in enclosure or exclosure studies where you have any of our native grazers, bison, elk, moose even, flame heights are much lower and spread rates are much slower because the fire fuel loads are reduced. It's especially powerful if you do a prescribed or cultural burn first and then introduce your native grazer. So the combination or pyrrhic herbivory, particularly useful. Elk are also important just because they're charismatic megafauna. And people will pay money to come see them. I'm not proposing we create a zoo, but at Oregon's Jewel Meadows Wildlife Area, they had a problem in winter. The elk would come down and depredate the neighboring alfalfa fields. And so they started buying alfalfa and putting bales on the refuge. And for $200,000 worth of feed, $6.5 million worth of parking and visitors fees were generated. So you could actually put a dollar value on wildlife viewing. I think it was 2011, a U.S. Fish and Wildlife Survey. California spent made about 11 billion in wildlife viewing versus about 1.2 billion on hunting fees. So a lot of money on wildlife viewing. At Point Reyes was where elk were restored around 1978. They are mowing the grass and changing the grasslands, taking out invasive. In Pacheco State Park, where just west of San Luis Reservoir, where they've reached great numbers now, you see bunch grasses coming back. And they're a missing prey item from much of our landscape. Wolves, bears, puma feed on them. Coyote take their calves. Elk carcasses are important for obligate scavengers, including condors. We still have supplemental feeding stations for condors because we don't have large ungulates roaming our landscape in California. But let's talk about fire. In this study, you see high fire fuel loads leading to a really big flame and no grazers, of course, and the tree will burn to the ground because the fine shrub will ignite the forest canopy and you'll get a canopy fire. On the bottom, in the browsed ecosystem, the crown does not ignite. The tree survives. The carbon stays sequestered because the grass is mowed and the shrubs are mowed. And this is happening at Point Reyes, right? The scrublands are becoming grasslands. Other places where there aren't elk, you see the Douglas fir re-encroaching. This is the 2018 California Department of Fish and Wildlife, CDFW, elk management plans map of our historic elk subspecies in California. In pinkish purple is Tule elk. These are all now, this is historical. They were hunted down to just 2 or 3 animals, which survived in the South San Joaquin Valley. Now we have 6,000. They are genetic siblings, right? They're all descended from 2 to 3 individuals. In the northwest, we have Roosevelt elk, or Rusi. They're the largest of the elk subspecies. And then the northeast, Rocky Mountain elk. The historic map in the 2018 plan omitted the Sierra Nevada as historic elk habitat. We proved that was wrong. And they are now recolonizing the northern Sierra Nevada with an elk herd at Stampede Reservoir, about 18 miles north of Tahoe and coming south. So I should have acknowledged the North Santa Clara County Resource Conservation District funded the habitat suitability analysis we did for Tule elk in the Bay Area, which will come up in a little bit. But we have elk in Santa Clara County because the Hewlett-Packard bought a ranch on Mount Hamilton, the San Felipe Ranch, and in 1978 worked with CDFW to get some Tule elk back. When they released them, they left immediately and went north to San Antonio Reservoir.
San Antonio Reservoir.
They went to the reservoir. Important to remember that. The next year they tried again, 1979. They went east there, Isabel Valley. There's some nice marshlands. They've gone a little further east since then, and now that we have an ecological reserve protecting the elk up in the San Antonio Valley. 1980, they try again, and they went to San Luis Reservoir. In 1981, they penned the elk on their ranch until they calved, and they stayed. And those elk, now often you see, if you know to look, as you drive south on 101, you look to your left, and they stayed. In the drought in recent years, 2 golf courses closed in southeast San Jose because they couldn't pay the water bills. And elk moved in literally within weeks, and they used the water holes on the golf courses. In the city limits of San Jose, we have elk herds. Now, where are the elk today? So the 2018 elk management plan shows in pink purple where the Tule elk have been restored. Basically, we have islands of elk, of Tule elk. The Roosevelt elk have moved back down and are now touching and admixing with the Tule elk on the coast. In the northeast, they thought these were Rocky Mountain elk that were starting to march south. Genetic studies by Ben Sachs at UC Davis, turns out these elk recolonized in the northern Sierra are about 3/4 Roosevelt elk and about 1/4 Rocky Mountain. So they're an admixture. Oops. And there was an elk herd in the Concord Naval Weapons Station in 1978. They relocated them to Grizzly Island. That herd. They relocated. One elk swam back to Contra Costa County. And they relocated him back to Grizzly Island. But look at this. Thousands of elk in William Heath Davis, this historical observer record, hundreds of elk crossing from Mare Island, basically the Napa side, across the Carquinez Strait to Contra Costa County. So they can swim long distances. They can cross the strait without difficulty. And as you think about wildlife linkages, don't forget you've got a big elk herd just north of you at Grizzly Island. But our highways are a problem. So courtesy of Professor Fraser Schilling at UC Davis and the Road Ecology Center, I asked him for a map of elk vehicle collisions, which would be the red dots. The green dots are black-tailed deer, and the brown dots are coyote, as you see Black-tailed deer are really stupid. They cross all the roads and they get hit all the time. But the elk don't cross the road. They don't cross 101, they don't cross 580, and they don't cross that little bit of 680. Connects and goes up and bisects the North Diablo Range. They're getting hit here at the Jaco Pass. I believe this is because here the traffic is intermittent, especially at night. It's high speed and intermittent, but on the interstates, it's relatively constant traffic, and so the elk aren't crossing. There's one red dot down there in Salinas, again, east of 101. So we can't get elk west of Highway 101 or north of Interstate 580. Although the Alameda County RCD is working diligently on wildlife over and under crossings for many species. But what are the yellow dots? Don't know. It's actually northwestern pond turtles. So, Thomas Connor, working with Justin Brashear's group here at Berkeley, did the habitat suitability analysis, looked at characteristics of San Luis Reservoir tule elk. Big herds there. They go back and forth. These little clouds are where existing tule elk herds are. As you see, none north of 580 nor west of 101. They are west of 101 way down in southern Monterey County because they were translocated to Fort Hunter Liggin, where they were poached to 0. But then they tried again, and that they now have hundreds of elk there. And we're looking at the characteristics of San Luis Reservoir elk, then predicting where is the good tule elk habitat. And son of a gun, the best part is Mount Diablo. But the Diablo Range, the Gablands, this big area of the Santa Cruz Mountains that has no dots is because they don't like the forest. They abhor redwood forest. Right? The San Juan, these tule elk survived from the Tule marshes. That's why we call them tule elk in the South San Joaquin Valley, and they have they don't like forest. Unlike Roosevelt elk, which love the redwood forest. But the thin strand along the coastal grasslands, the inland side of the Santa Cruz Mountains, the East Bay Hills, and the rest of the Diablo Range, all good elk habitat. They go back and forth in Santa Clara County from San Luis Reservoir, which is Santa Clara Merced, to Anderson Reservoir, Creek, south of San Jose. And the next slide shows even more vividly. The greatest concentration is north of Interstate 580. It is the North Diablo Range, habitat quality-wise. In Santa Clara County, they go to the reservoirs because they love the green up. In the late summer and fall, the grasses become desiccated, but the reservoirs are receding, and forbs and grasses are coming up, and they love that green up, and they follow the reservoirs as they recede. That's important because when they do cross 580, where are they going to go? They're going to go to Las Vaqueras, I predict. So you have a patchwork. This is a map of conserved lands. And the Alameda County RCD is working on crossings that will enable them to hopefully reach East Bay Hills. They'll have a couple barriers, perhaps 680 and 580, and then 580 again. But I think really important, if we don't have the species back, our wildfire problems will be worse. One of our publications worked with Tom Batter, California Fish and Wildlife, elk and pronghorn specialist, and many other academic and wildlife biologists that study elk, to show the elk go under roads. A lot of people say, oh, elk like only to go over, you have to build these expensive overcrossings. No, they go under. This is a medium-sized bridge. We used to calculate the openness ratio, the height versus the width and the depth or the length under the bridge. And it is probably true that bigger is better for elk to go under, but the real characteristic we found in all of the elk undercrossings that we recorded, all 3 California elk subspecies go under roads. The common characteristic was this, an intermittent stream. They love to follow these as game trails, apparently. And the smallest opening was this culvert at Fort Hunter Liggett. The red arrow is pointing at the trail cam. We have video of a herd of elk going through this. The bull is like, trying not to scratch his hair on the roof. I don't think I don't know why they did that. I mean, they can jump an 8-foot fence. And this barbed wire fence doesn't even go that far on the 2 sides. They can just go around, but they follow this little spindly trail and went through. So bigger is probably better, but presence of an intermittent stream should be factored into your thinking about wildlife linkage under crossings. Elk can cause problems. They can break fences. Bull elk can do a belly flop on a barbed wire fence with impunity and flatten it for the rest of the herd. When they frequently break a fence in the same spot, you can put in these H-frame style wooden crossings where it's lower. The top horizontal board might be 30 inches as opposed to 40-inch high barbed wire, and it probably works half the time. You can see the herd of elk in the background there. This is Mendocino County. They have private elk hunts now. $20,000 for a bull, for a tag to hunt a bull. $5,000 for a cow, because those cows, elk cows, don't have antlers, so it's a premium. But generating money. 3 of my 5 sons live in Montana. This is Gardner, Montana. I think it's unusual. Normally elk are shy of humans, but this is just north of Yellowstone, and they have acclimated to the Gardner High School football team, and they sit in the end zone. So a lot of people ask me, are they going to eat in my garden? And in Missoula, where my boys live, they do have elk in the city, but it's the deer that are eating their gardens, not the elk. But I hope you'll share my enthusiasm. Thank you for the opportunity to speak.
Thank you, Rick. Any questions?
First of all, go Bears, beat Stanford. Next year. That was pretty interesting for a Stanford grad. I'm curious. I am curious about the genetic diversity. It started with a small group of 2 or something you said. That's pretty dangerous country.
It is. It is. So we have elk with antlers coming out of their eyebrows. We call them unicorn elk. The Thule elk are ridiculous. It's amazing they've done as well as they have. We're up to about 6,000 Thule elk, about 6,000 Roosevelt. I don't know. But Ben Sack's genetic study just a year or 2 years ago showed that they're admixing everywhere. In Fort Tejon, right on Interstate 5, you should go pick grapevine. That rancher got some elk from greater Yellowstone area. Those are Rocky Mountain elk. They broke the fences. They're colonizing the Tehachapi's and up in the southern Sierra now, up to Lake Isabella. But one of them crossed Interstate 5 and is in the Windwells Preserve on the west side, from the east side to the west side. One bull crossed and is in the middle of this big Thule elk, has a harem of female Thule elk. So in San Simeon, William the First brought Roosevelt elk down and released them. And although the Thule elk at Fort Hunter Liggett won't cross through the redwoods in the Coast Range to go to the delicious grasslands, those Roosevelt elk would cross through and are now mixing inland with the Thule elk. So we're getting genetic diversity whether we planned on it or not, because we made this gamish of elk subspecies.
So
My family is up in we're right where the Tule Elk herd is at the northeasternmost point, so kind of where 580 does the corner up between Tracy and Livermore. And so when I was growing up, the Tule Elk were much more higher in the foothills. We saw them a lot in the foothills. They ranged further south that we saw. And that herd now mostly sits down in the valley floor, really doesn't range far south, doesn't go up into the foothills. Have there been kind of any observations about changes in behavior over like decades with the Tule Elk where they're actually not ranging as far? Or is this just kind of unique to a particular herd, or any information about that?
I think it's because we have a lot of poaching. And not far from that Hewlett-Packard Ranch, there's an elk graveyard. And it's all these elk skeletons, but no antlers. So I think there's a lot of poaching. I don't think it's the ranchers. I think it's city people that go out and poach. We see people chasing elk with ATVs. So I think these factors have make it probably almost impossible to figure out the answer to the question. There's so much human interference now. In Missoula, as you see, you could walk right up to an elk. In Santa Clara County, if they see you from 5 miles away, they run the opposite. They'll disappear to the very next ridge before you even get a chance. And I think it's because of human interference is driving a lot of these funny behaviors.
So in Point Reyes, as they take the cattle off, and there's concern that the coyote brush is going to remove the grassland there, so you're feeling that the Tule elk are actually going to, do they graze on and keep out the coyote brush, or is it expected to take over?
If you go to Tomales Point, where they were fenced in by that 12-foot fence until recently, it's just lupins and grasslands and forbs. And then they established done a lemon tour. They brought some before they breached the fence, and same thing's happening. The scrub is going away. It's becoming grasslands and forbs. When they eat bunch grass, they don't chew it to the ground like sheep. They don't uproot it like cattle might when it gets moist. They'll uproot and eat the roots. Our Tule elk co-evolve with the bunch grasses. So they nip it and don't kill it, and the root system goes to about 4 times bigger because they're pruning it, sequestering a huge amount of carbon versus bunch grass that isn't grazed by elk, but the root system's much smaller. So I think we just have to put the right species with the right species of plant and animal to get the greatest benefits.
Thank you very much. Very interesting.
I'm going to resist from commenting on the Stanford-Cal rivalry, as I think most people know where my sympathies lie. I really have nothing technical to add. I will add one anecdote to what Rick was talking about. When my wife and I went to Yellowstone for the first time, we drove up into the Mammoth Hot Springs area, and on the lawn by the Mammoth Hotel were probably a dozen or more elk with one big bull, and we thought they were statues. I mean, literally, the parking lot is here, the lawn is here, the hotel's here, and they were right there on the lawn with people all over the place. We thought they were statues, because how could elk be right there with all these people? But heck, just like the football picture. What I want to do is, first off, say I think the presentations today were great, and want to thank you all for doing that. This program has grown tremendously from a very small room over in MVZ, sitting around a table, to where we are today. And just look at how many people. We've filled the room. And I think that speaks to the quality of the presentations and the work that Save Mount Diablo and the Bowerman Committee is doing. So with that, I want to thank first off our staff who put this together. They work really hard on it, do a very nice job. You get better food here than most places, I'll tell you. Not just the organizing the colloquium. So thank you very much for that. We have a committee, the Mary Bowerman Science and Research Committee. We do a number of things, and the members of the committee, last time I botched the name of one of the committee members, so I'm going to avoid that. But on the back of your program is a list of all the committee members, many of which are here today. So thank you for that. So that really summarizes what I had to say. I've enjoyed it. Look forward to next year. Oh, one more point is we sponsor these research projects. We don't put a whole lot of money into them, quite honestly, but we do hope that it results in other grant funding that go to them. And we've already got 5 proposals for next year. And I think that speaks to the success of the program. Okay, well, thank you all very much for coming and enjoy your lunch.